Good morning, one and all. So let's start today's proceedings. So this is the fourth day of our workshop, and today we'll have three sessions planned. One is on Introduction to Oncogenomics by Dr. Abhishek. Second session is on Eliminating the microRNAs by Dr. Ravi Shankar. And then third lecture session will be on matrix methods in data science by Dr. Dilpreet Kaur. So let us start. Let me introduce you our first speaker of the session, Dr. Abhishek Kumar. Dr. Abhishek Kumar will deliver a talk on Introduction to Oncogenomics today. Dr. Abhishek is a senior bioinformatics and genomics expert with over 20 years of experience. He received his PhD degree in bioinformatics from University of Bielefeld in Germany. Further, he worked as a postdoctoral fellow in the Department of Biology at University of Padova in Italy, where he established pipelines for pharmaceutical company Roche 454-based transcriptome analysis for insects and fishes. Thereafter, he moved to the Institute of Botany at the University of Kiel in Germany, where he established multi-omics approaches for characterizing the future drug-like compounds, also to deduced biosynthetic gene clusters of marine fungi, and studied fungal adaptations. Currently, he has, to his credit, sequencing of the largest number of marine fungal genomes in the public domain. And later he moved to the German Cancer Research Center, DKFZ in Heidelberg in Germany, where he worked on familial cancer genomics and developed interesting pipelines for analyzing the whole genome and whole genome sequencing data and analysis of more than 300 samples of various cancer types. So I'm glad that he joined us. He has agreed to give a talk. Unfortunately, due to some emergency travel, he will not be available in person live today. But he agreed that he will be joining a bit later to address the questions and to interact with the attendees. So unfortunately, we have only the video for the beginning. So we will be playing a video, and that is a recorded lecture by Dr. Abhishek. And then later he will be joining, so you can ask questions and you can interact with him, hopefully, when he get a little bit of time. So, so without further delay, let us start. So now I request technical team to share the video lecture. Thank you so much, Dr. Abhishek. So it's a pleasure that you are here now. So Dr. Abhishek
Ye-
has joined us live.
Thank you.
So if anybody has a question, please feel free to write in the chat box, or you can also raise your hand so that he can address you directly. You can also have an interaction with you.
Mm-hmm. So looks like there is no question, or I'm missing something.
Okay, seems the talk was very much clear. Yeah, I think we have we have Rajesh Anand who want to interact with you. So please go ahead.
Okay.
Rajesh Anand, please, you can ask, you can unmute yourself and you can interact with Dr. Kumar.
Hey, can you hear me? Good morning.
Good morning. Good morning.
Yes, sir. I have a couple of questions. I like to first one, how to validate all the specific circulating genomics or like RNA in blood? Because I'm interested to work in colorectal cancer. So
Uh-huh.
I wanted to know how to validate those genomics because initially I had planned to work with the proteomics, but it's really so vast.
Okay. So
Yes, sir.
what you can do is that you can do the transcriptomics of the blood
Mhm.
in two different conditions, and actually there is a pipeline. Uh, there is a method called uh, microRNA-seq. Yeah. So it will, from transcriptome, you can actually all detect all microRNA from the sequencing data,
Okay.
and uh, uh, and then you can merge with the existing human microRNA data available in Miranda.
Okay. Yeah, actually,
Uh,
I am aware of that Miranda is more suitable for metabolomics, I think.
mhm.
But actually, again, the microRNA, there is a small issue. Thing is, all the data that exist in the public repository is not really validated, so a bit of scary to use those data. So I'm planning instead go for RNA sequencing like transcriptomics something. So
Yeah,
is it meaningful or is it worth the cost effective than going for
it's a
proteomics?
cost effective actually. So we have seen it, we have done it, and that is for some projects
Okay.
where people have less money.
Mhm.
And so basically, and not just less money, you will identify microRNA,
Okay.
and these microRNAs, since human annotation is available, so
Mhm.
there is a group which has developed a tool for microRNA seq, based on Illumina, and this tool is really easy to use because
Okay.
it requires a group from Berlin. I remember, if I remember, Max Planck Berlin has
Okay.
developed this tool. And I'm looking. Yeah, so this tool is called
Is it
uh,
group from
Mir-
Vladimir from University of Heidelberg, Vladimir?
no, it's a Mir Deep 2.
Okay, okay, okay.
Yeah, so it's from MDC. I'm sending you the link. So
Okay,
I actually personally have run it several times, so we have a good
okay,
success.
okay, great. Great.
And the price wise also it will be effective uh,
Okay.
for you. So I am just sending you the link to everyone.
Sir,
So
I
that
have one question.
yeah.
I would like to study some differential expression of proteins and RNA gene transcripts at IOB, your Institute of Bioinformatics.
Yes.
So could you please help me? How can I do that? Or can you share me your email so that I can send you in detail?
So
Of
we
course,
have a
I already
proteomic
prepared.
service. So you go to our website and write for proteomic service. Okay.
Okay.
And they will
Actually, I haven't got any response at the time, but I was scared whether that because of the COVID and all, they.
yeah, there because of COVID, we have some structural change. At the moment, we are not accepting the external projects. Yeah.
Okay.
So, uh, mm, uh, but if you come up, you can write me, and if you come up with a strong argument that why you want to
Yes, yes,
do
yes,
it,
yes, please.
we
Can you share
will
me your
we will
email, please,
definitely
so that I can.
consider, and then we can actually discuss with our team. Uh,
Okay.
and like we do not want to give a commitment and do not follow it. Yeah. So
Yeah.
that's that's
Can you
the
share me your email, please, so that I can email you in detail?
yeah. You will get my email ID. I'm sending you. Yeah.
Yeah, yeah, perfect, perfect. Okay, okay, that's it. I have only these questions. Thank you so much.
Yeah. I'm sending it to you. Okay.
Dr. Kumar. So let us see if there are any other questions. It does not seem that we have any further questions. So let us thank again. So we will now wait until our next session starts at 11:30. So we can have a pause for 5 minutes and then we will resume. Thank you, Dr. Abhishek Kumar.
Thank you. Thank you. Have a nice time.
And you too, sir. Thank you.
Hello, good morning, sir.
हेलो
Sir, you are audible.
आगे
Yes, sir.
हेलो
Yes, sir, you are audible.
लेकिन वो नहीं आई ना, सुनने वाली नहीं। हाँ अच्छा, मेरा ऑडियो गया सर। उन
Can you hear me, sir?
ठीक ठीक, वो बोल दिया ना, वी कैन हियर यू। हाँ बट सर इनको वो कुछ बोलेंगे तब सुनने का देख। हेलो
Hello, sir.
हेलो,
Yes, sir, you are
आ रहा है
audible.
उनका? नहीं, आवाज नहीं आ रहा है इनका।
Can you hear me, sir?
Yes,
Hello.
Rajveer, we can hear you. It seems there is some issue at the speaker setting
वो
of
वाला देख रहा है।
Yes, sir.
Dr. Ravi's side. So maybe they will fix it.
इसमें
Please
स्लाइड
wait for a
क्योंकि
minute.
अब ये सारा हेवायर हो गया। अरे हेवायर, हेलो, इसमें आई उनकी आवाज लेकिन अभी आ नहीं रही है। सिस्टम को ऑडियो तो आ रहा है,
हेलो
सिस्टम को ऑडियो आ रहा है
Yes,
लेकिन
sir, you
मन
are
नहीं
audible.
सुन रहा है। मेरा वहां से ना ये ले आओ, लैपटॉप ले आओ, सिस्टम को ऑडियो आ रहा है, उनका आवाज नहीं, ये म्यूट करा हुआ है ना उन्होंने से। तो कुछ जब बोलेंगे तभी पता लगेगा। तो हमको तो उनका सेशन दिखना चाहिए था ना। सेशन तो जरूर उन्होंने चालू नहीं किया, ये दिख रहे हैं ना सब जो भी लोग वो हैं वहाँ पर। वो सेशन जो चालू हो वो दिखेगा, बाकी ये इसमें हम यहाँ ज्वाइन कर लिए हैं, यहाँ से आवाज में जा रही कि नहीं जा रही पता नहीं चल रहा है। आवाज तो यहाँ से जा ही नहीं रही इसकी, कम से कम होनी चाहिए थी ना आवाज यहाँ से।
Sir, you are audible.
हेलो,
Yes, sir, you are audible.
आवाज आ रही है मेरी?
Yes, sir.
आप लोग की आवाज नहीं आ रही मुझे। ना कोई इवेंट पे स्क्रीन पे कुछ आ रहा है, कुछ शेयर नहीं हो रहा अभी, वहाँ इवेंट नहीं चल रहा है क्या?
Sir, you are audible and your video is on.
नहीं, मेरी आवाज आ रही होगी लेकिन आप लोग की आवाज इस पे नहीं आ रही, और ना कोई ये हो रहा है, क्या कहते हैं उसको, इवेंट का कुछ दिख रहा है वहाँ पे।
Sir, you are the presenter. You have to share the screen.
मैं स्लाइड शेयर करूँ?
Yes, sir.
यहाँ से कर दो, पेन ड्राइव से कर दो। एक मेरा दूसरा लॉगिन भी, 2 लॉगिन है ना, उसमें से आप एक काम करेंगे, दूसरा लॉगिन भी मेरा जो है रविशंकर के नाम से 2 लॉगिन होगा, दोनों को आप ये कर दीजिए ना। ओके सर, है ना, दोनों को ऑथराइज कर दीजिए शेयर करने के लिए। यहाँ हो रहा है, उसको नहीं कर सकते, उसको नहीं कर सकते, नहीं हो पाएगा। ठीक है, तो मैं तब यहीं से करता हूँ। ठीक, चलो सो इनकी आवाज का कैसे वो होगा? उनको उनकी आवाज का अब यहाँ से हम देखते हैं जो रास्ता इसी में चलाएंगे। ठीक है, हम लोग स्टार्ट करते हैं। आवाज आ रही है मेरी ना क्लियर आपको?
यस सर।
ठीक ठीक ठीक। So a very good morning to all of you, and I'm going to present work here. This is actually summation of work done by us in last 10 years, something like that. In 2010, we had started this lab, and with starting of this lab, the microRNAs were one of the most important components. Okay, just a minute, so chhota karo. So the title today I have kept is Ruminating MicroRNAs Computationally Decoding the Ultra Regulators of the Living System. Yeah. So in our lab, we actually work in multiple areas. 5 major areas we have identified. That the first one is regulatory and RNA biology. Then second is about RNA binding proteins. Third is computational genomics. The fourth is next generation sequencing, and the fifth one is next generation computational solutions, like developing things on GPU and high performance computing. So out of these, I have been asked to present work on microRNAs, and here it is. So a living system, आप लोग में से किसी को भी कोई दिक्कत होगा तो मुझसे उसी वक्त क्वेश्चन पूछ लीजिएगा। हिंदी में भी पूछ सकते हो, इंग्लिश में भी, कहाँ जहाँ पे भी होगा, जिस तरह से कंफर्ट लेवल आप होगा, आप मुझसे क्वेश्चन पूछ लेना। ठीक है, आप लोग की किसी की आवाज़ वैसे मुझे आ नहीं रही है यहाँ पे। सो एक जो इन द लिविंग सिस्टम, the cell system is controlled by this particular composition, like that it has got DNA, and from DNA RNA is transcribed, then RNA is translated into proteins. So those RNAs are called coding RNAs, and those who don't translate into proteins, they are called non-coding RNAs. So major emphasis so far has been upon the coding RNAs, which code for proteins, and total neglect of non-coding RNAs happened. But actually the non-coding RNAs work like kingmakers. So coding just, they, as you know that in this human cell system, there are just 3% coding genes and rest are non-coding elements. Okay, and one of them are microRNAs here. They are 21 bases long. They exist mainly in duplex form, as you can see here. They exist in a duplex form, and just like mRNAs, they are also transcribed like pre-microRNA. Here you can see that this pre-microRNA form, it is longer. Then pre-microRNA is further processed by two kinds of RNase enzymes, DOSA and DISA system. So here is a DOSA system which acts on it inside the nucleus in animal system. In plants, both of them work together. So in animal system, the DOSA nicks the extra region, and you can see here that the paired region is retained with terminal loop. This is called terminal loop, and this is the paired region. And 3' overhang is there after nicking of those duplex regions. So this duplex overhang, 3' overhang, is the one of the characteristic features of microRNAs. Then this microRNA is exported out of the nucleus in the cytoplasm. You see here that in cytoplasm, then DISA recognizes this microRNA with 3' overhang system here. Then DISA again chops it, and just like two combs attached together with little bit of 3' overhang from both sides, this duplex is formed like that. One of this duplex partner is then associated into this risk RNA-induced silencing complex. We call it RNA-induced silencing complex. So one of these partners is taken by this complex, and what this complex does, this complex, this one attaches this one of the microRNA partner to the RNA to which it has target. Okay,
So what happens now that once this small RNA joins this messenger RNA, it stops, it halts its process of translation. Translation is stopped, and gradually then I have written here that translation inhibition, two effects are there, translation inhibition or transcript breakage. But once translation is inhibited, the transcript is broken into small pieces. It gets degraded actually. So end of the day, it is always transcript breakage. Translation inhibition is always followed by, because without protein, RNA can't survive. So for binding, you need some protein. So no RNA can survive without RNA binding proteins. So here in this case, translating proteins and those proteins which were binding to the RNA were associated, and as soon as microRNA came, that association went away, and this mRNA gets degraded. So this is the mechanism which has been highly ignored for a long time. Later on, by the mid of 2000, this starting decade, like around 2004, 2005, it was realized that no, they are going to be a major regulator. So at that time, they found that these microRNAs, they regulate almost 1/3 of the genes in any given system. At that time, only 700 microRNAs were recognized. But by now in human alone, more than 2000 microRNAs are recognized. And if you go through their analytical data, you will find that almost every gene is getting regulated by this microRNA. So our work has been mainly focused upon these microRNAs, how they are being formed. So in last 10, 12 years, we have done extensive studies of these microRNAs, how they are regulating. Earlier, people used to focus regulation of gene upon those transcriptional regulation, like how a particular promoter is activated and then how a gene is transcribed. But after microRNA, the whole concept has got revised again, and more emphasis now being done on post-transcriptional regulation, because at transcriptional regulation, only 3% these transcription factors express themselves, and very limited control is done by them actually. Okay, but major control system acts at post-transcription level, and microRNAs are one of those major component post-transcriptional regulators, and it is mainly inhibitory in nature. So when you have to stop some gene, the microRNA chips in, and it does its job. Expression of gene is halted, and then another set of genes are promoted. Then again at some time they are halted. So this braking system, post-transcriptional braking system, is provided by these microRNAs. They are very much critical for regulation system. Second thing is that they are highly spatiotemporal. Spatiotemporal means that they are very specific to moments, how much time they have to spend for a particular phase of cell development, and when they have to vanish away, and when they have to start the thing again. So this entire combination of microRNA, various kind of microRNAs, decide how the cell is going to grow and in which phase it is getting into. In any aberration in this process, entire process, it results into some kind of disease or certain abnormalities. So I share a timeline from a very great paper. It is a review literature actually by Eric Lai in Current Biology 2003. So he has given this complete timeline of microRNAs biology development. So as you can see here that microRNAs were first found here, but the finders were not able to recognize them as a microRNA as such. Later on, it was found that this Lin4 microRNA is here, and its target is Lin14. So what happened is this work was done in C. elegans, and their cell development was found getting affected. And when they looked into, they found that there was some allele in the genomic location where if mutation happened here in Lin4 region of that genome, then Lin14 gene was out of control and it was expressing it very much. Then they looked into the intronic region of this gene, sorry, in the genomic region, and they found that it was coming from an intronic region, which was not a protein or a coding region. And they were stunned. Thereafter, they do some PCR experiments and all these things, and they finally found that a small RNA interference system was discovered, and same kind of small RNAs were found from many locusts. So what happened that thereafter they formally recognized them as the regulator, major regulator, and extensive work was done in first worms, and after worms the further study was done into other species, and finally they found that this particular small RNA of length of 21 to 22 bases, they were present in almost every eukaryotic system, and they were regulating the genes in such similar manner, in post-transcription manner. They bind to the messenger RNA and they halt the process of translation. And this way, this entire field has developed a full-fledged form. And now, since you must be stunned that who is this guy. So as I showed you previously, that microRNAs, they exist like this. There is a terminal loop, and then there is a stem where pairing happens, and there is a 3' overhang. This is further processed into mature microRNA form. So first form is called primary microRNA, which is just raw transcript from the genome, and then from this primary microRNA, pre-microRNA, which is called pre because of it being the precursor. So precursor microRNA is formed, and then from precursor microRNA, the mature microRNA is formed. So anywhere if you have to find microRNA, then you have to first find out the precursor microRNA, if precursor hairpin is forming or not. Interesting thing is that in your living system, no RNA exists in a linear form, what we are actually taught in books, because in books they just show you that linear line, something straight linear line, and they represent it as a RNA. But no RNA can actually survive without folding. These folding is like that. The terminal loop is there where no pairing happens between the bases, and here self pairing happens. So due to self pairing, this stem region is formed, and then unpaired region is again there, which is in microRNA case is nicked, and finally the paired region is retained. From this paired region, one of the strand goes into the target region of messenger RNA to halt the process of translation and degrades the messenger RNA. So very important thing is the secondary structure of the RNA, how it is formed. So far the computational methods, they focus upon the folding pattern of these RNAs. These folding patterns, they were studied for their minimum fold energy. You must be knowing that free energy is something which is measured for molecular stability. So if the free energy is very high, it is not going to be a very stable thing. And if the free energy is lesser, then it is going to be stable. So when we analyze it, we find that there is lots of fluctuation in the free energy actually of the fold. The minimum free energy range, you can see here that 170 kilocalorie, minus 170 kilocalorie to minus 30 kilocalorie. Okay, and average is 0.76. So dispersion is very high. And up to this time, the computational biologists, they have been using only minimum free energy has been one of the major factors to determine whether the folding belongs to messenger RNA or something, microRNA or something else. Similarly, the loop size, this terminal loop size, it varies from, you can see, 4 to 94 with average of 43 bases. So you can, even this region, the terminal loop region is varying too much. Mismatches in this region, the paired stem region, you can see here that 0 to 25, 100% match to 25, and average is 7. The stem length, this one is called a stem length, is 53 to 98. So you can finally see that lots of variation is there, and if you look into these factors alone, lots of misclassification of microRNA happened, and this was found actually. So if you look into previous developed software, lots of error they were doing in recognizing, a lots of positive, false positive. False positive means that they were not microRNA precursors, but they were recognized as microRNA precursor because the initial studies were done basically on these factors. These values were being used, and based on this, microRNAs were being screened. I share with you a very important factor, a fact that if you scan the whole genome itself and you look for the same kind of hairpin loop structure formation, almost entire genome you will find studded with this kind of hairpin loop structure. So this problem is going to persist, and this was happening with most of the computational biologists. They were unable to decipher that what to do now. So we started working on this part, and there were certain realizations with which we were working, that poor implementation of microRNA biogenesis models was there. Most of the existing tools have concentrated on canonical microRNAs. Canonical means that microRNAs which follow this particular path which I showed you, that this particular primary is formed, then primary is processed by Drosha RNase enzyme, then this precursor comes out, precursor is processed by dicer, another RNase enzyme, and then mature microRNA is formed. So this is called canonical pathway. This is a very typical pathway. And very interestingly, I will be coming to a slide where I will be showing that these all enzymes are expressed all the time in the cell system, but microRNA composition varies a lot. So this traditional approach was wrong itself. However, many canonical microRNA are now being reported. MicroRNAs differ a lot in their sequence composition, hence tools need to make better use of sequence information. If you look for just homology, then you will end up only finding similar kind of microRNA. You will not able to detect novel microRNAs. Similarly, thermodynamics and structural parameters are weak discriminators. Many non-microRNAs overlap with true microRNAs for their parameters. So as I showed you here, these values, if you look for even non-microRNA region, they share lots of similarities. That's why lots of false positives were being predicted. So we realized that true understanding of microRNA's properties was necessary. Innovative computational approaches were required, which were technology driven. So technology driven now becomes important. But what technology? I will be coming to that part very soon. So here is the first technology. You can see here that MIR-base is a database where all microRNAs are and the known microRNAs are archived, reported there. This is the official database where this information is collected. And as you can find here that microRNA information has grown with time. Okay, with every new release, it has increased a lot, significantly. Actually, it is not a linear kind of, but it is taking almost exponential shape actually. And here in the parallel, I show you this slide here, the why all of a sudden this jump has happened. So in 2005, actually, this particular thing, just a minute. In 2005, this in around 2005 and 2004, you can see here that next generation sequencing was introduced. So earlier, using this PCR or all these stuffs, microRNAs were being reported. So that was very slow process. But with introduction of next generation sequencing, it was possible to sequence even a single copy of RNA. So what happened here, that Roche system came and little bit increment in the number happened, and thereafter, as you can see here, Illumina arrived. As soon as Illumina arrived, entire scenario changed. Illumina sequencer is another sort of short read sequencing platform, and due to that, lots of revolution has happened in biology, and microRNA is one of them. So lots of those regions which were never studied, and it was also not possible to do in PCR for every region. So what happened that this impacted a lot, and that translated into the reporting of lots of microRNAs actually. So how NGS boost microRNA discovery? So I told you that precursor regions are formed, the pairing between, the self-pairing between the various part of the RNA is studied, hairpin loop, terminal loop is studied, stem length is studied, or minimum fold energy is studied, but these all properties are well shared by other non-microRNA region also. But once you get small RNA sequencing reads here, these reads are mapped to the regions, okay, and they support the formation of microRNA theory for that given precursor. So here you can see that if you get support enough for read mapping here, then you say that yes, this precursor is going to work as microRNA. So it in some way guides your microRNA formation. So what we did that after realizing all these developments, we also found that so far the parameters being used earlier by researchers were not sufficient. So we introduced three things, and we developed new software MIRBAG, a novel approach for microRNA identification, where we applied machine learning system, ensemble machine learning system, where you can see that three different machine learning systems were combined as a jury. Okay, the jury would decide whether this region is going to be a microRNA region or not. So support vector machine was applied, naive Bayes was applied, and best first decision trees were applied here. And this made our ensemble machine learning system. But only what kind of system you are going to use, it never going to work if you mindlessly provide the input to that system, because you need to derive good features, important features, significant feature. We felt that this entire microRNA folding structure system, okay, I just show you one thing here, that how you get the fold, because that is the important thing. Just Google it here. This is a web server actually. So I just show you here that example. So this RNA sequence I am giving here, and I just run it. These parameters are default parameters. I'm just going to show you how this one is predicted into a structure by a software. So you can see that you had provided this particular RNA sequence. This RNA sequence is folded into secondary structure. So function of any RNA is not dependent on its tertiary form actually. Tertiary form is very transient one. It keeps on changing. Slight of temperature, it changes a lot. What holds the key is the secondary structure. That's why everything is focused upon secondary structure. So this is the secondary structure form, which I had shown you there also. This way it is folded, and one other representation is dot bracket form. So these dots represent the loop region, this region where unpaired things are happening, and left side, like left side opening bracket, is like this base is pairing with this base at this position, corresponding position. So this entire structure is actually resolved in a dot bracket form. And I tell you one thing, that if you take dot bracket form for very long sequence or longer sequence, then it does not work actually. So WebEx कहाँ चल रहा है हमारा नितेश? मैं कहाँ पे खोल के रखा हूँ? समय यही था.
हाँ, इसमें लास्ट वाला होगा.
So folding, you can see there that this same thing was converted into dot bracket form, and we realized one more thing, that with our previous work, that dinucleotide density actually captures better information of secondary structure than these folding programs, especially when the length is longer. So somewhere we were getting discouraged of using these de novo structure prediction programs, but we took input in these forms. 4 mononucleotide ATGC were considered. In RNA, you consider uracil instead of T. So they are almost like synonymous for us. 16 dinucleotide compositions were considered, and 20 structural triplets were considered from dot bracket representation. And normalized sequence based total pairing. So how much pairing was happening to, in a given window, we were that were considered. And you can see that from this region, we found that 21 base pair window was working as a good window. So we kept on sliding the window for the whole length of this RNA, and accordingly we calculated these features on this particular window size. And how we reach to 21 base pair window, that we do by randomly trying and checking which one was giving best feature separation between the positive data set and negative data set. The positive data set is our microRNA precursor, known microRNA experimentally validated sequences, and negative data sets were those regions like tRNA, mRNA, or other RNAs which were not microRNAs, and they were experimentally validated. We find that F score, the feature scoring system, which were having strong discrimination between these two sets, were best at 21 base pair long sliding window. So these 4 properties were considered on this 21 base pair sliding window, and another thing was full length based properties. So full length based property are consecutive paired bases, that how much longer the longest consecutive base pairing was happening. First thing. Second thing was structure profile scoring. Works in sliding window whose size is determined dynamically by the length of the profile being scanned. So profile being scanned, I come to that part. So these properties were fed into form of feature vectors, and these feature vectors were finally given to the machine learning system for training, and based on this machine learning training system, we identified precursor microRNA with very high accuracy. So this about this thing I was telling you, structure profiling scoring. Mature microRNA, you must be knowing that dot bracket are so much simplified form, and if you try to find motif of any structural pattern, okay, that fails actually, because dot bracket, they keep coming like that. So what we did, that we enforced mature microRNA region in all these precursors, and based on that enforced mature microRNA precursor region, we performed multiple sequence alignment, and we restricted 80% similarity and formed different clusters which were having similar kind of alignment. And these clusters were formed, you can say that they are structural families of microRNA precursors. Accordingly, a pool was prepared, and then every pool was having a consensus structure. So all these families were having some sort of their consensus structure constrained by mature microRNA region. So after that, once you input any sequence for scanning, it is scanned against all these pool members, and whichever member returns the highest scoring system, that pool is particular structural profile from that family is considered and given input to the machine learning system. So we found that all these worked very well, and very high accuracy we obtained consistently, and especially this structural matrix style which we developed, it was consistently working among the best features for almost all the species here. As you can see, the rank is almost in the top ranks. So we compared, that is very important that you do benchmarking also, that how your software is performing compared to other software. And we found that the benchmarking, when we did that, MIRBAG, as you can see here, that MIRBAG was attaining very high accuracy. Okay, and it is not just accuracy which matters, because at one side it can become 100%, and on other side it can become 70%, 60%. So sensitivity and specificity balance is very important. What is sensitivity? Sensitivity is the fraction of correctly identified true positives. Okay. So how many micro out of the entire collection of known microRNAs, how much correctly you identified the fraction of correct microRNAs? That is measured by sensitivity. Specificity measures oppositely for the negative data set part. So those which were not microRNAs, how correctly they were identified, that is measured. You can see here that the balance between these two is the best, almost best among all these compared softwares. And Maybach was not just having highest accuracy, but also best balance it was providing, which was possible due to ensemble nature. You see this ensemble nature of machine learning. The ensemble approach, wherever you apply, they assure you the balance between sensitivity and specificity. Okay, so now we were done with this part, yet we were getting challenge here, that you can see here the Mr. microRNA is still challenging us, and it was telling us that despite of all these things, still you are not able to recognize me, and I will keep on evading. So here comes the realization why I said so and why Mr. microRNA was telling like that. So Mr. microRNA was telling like that because if you look into the databases, there you get a big truth that most of the reported microRNAs were from the species whose genome was sequenced. So even in my case here, as you can see that I take a sequence, long sequence, because precursor you can't tell like that. For precursor you need to scan genome. From genome then you take region of certain size and you fold them, derive a secondary structure form or something like that. Some way you have to recognize the precursor thing. Without precursor you can't work, and a precursor can't be recognized without the reference genome. So that means that if you don't have a genome of the species, then you are not going to do its microRNA biology, as simple as that. And that was pathetic thing, because most of the species, plant species on which we work here also, their genome is not sequenced. For very few at that time, when we had done that work, at that time, almost 84% of the microRNAs were from the sequence genome. They were coming from the sequence genome. And just 16% of this was coming from those species whose genome were not sequenced. And even those 16%, they were identified through homology search, similarity search. उसको देखा कि वो कितना सिमिलर उसके साथ दूसरे जो स्पीशीज में रिपोर्टेड है उससे हो रहा है, उससे लेके इसको रिपोर्ट किया. And it was so much pathetic situation, and NGS had not picked up that much even at that time. So we realized it, and we thought that what if we develop a software which could work without the reference genome, where you don't need to provide the reference genome and just doing small RNA sequencing data, because the small RNA sequencing cost is much cheaper than genome project. Genome projects are very much costly. So if 1 lakh, 2 lakh rupees mein, if you can do that thing, that is very big, and you are out of that bottleneck actually, and you can do microRNA biology. So we started looking into the pattern of pairing between these two mature microRNA strand regions. So NGS, from NGS data we get this small RNA sequence. Now we have to just validate whether they are partners or not. So we developed a Knuth-Morris-Pratt based algorithm based search system and implemented it into HMM form. You can say sort of thing, HMM sort, because why I'm telling a hidden Markov model form, because it is just like that different kinds of states where instead of focusing on sequences, what we focused upon was deletion pattern, mismatch pattern, matching pattern, and insertion pattern. So insertion deletion, like if this region is not pairing with this region, then it is insertion. If this region is not pairing with this region, then it is deletion. Okay, bulge, something like that. matching region is like here it is pairing with this one. It is bulge deletion on both side, you can say it. So like that we recognize that 4 major states were there, and the pairing between these states for this entire length was important. So if duplex length is 21 base pair or 22 base pair long, it is, then accordingly what I started looking into that if first position is having match, okay, then how it is associated with mismatch or deletion. So you can just see here, like I'm showing the matrix here, the matrix position, the position wise probability that at first position it is there, and at second first position it is there, and second position some mismatch is there. So mismatch combination, the mismatch deletion combination, mismatch insertion combination, and insertion combination, and deletion combination, how they were distributed across the duplex. These were revealed by this matrix. So same matrix we used and trained the machine learning system on this entire thing. And we developed a tool, MyReader. So MyReader is there on our website. You can download it and work with it. So the MyReader was trained, and same way you can see here that we used this particular system to reveal this what you say that mature microRNA strand from small read. So here you get pool of read data here, small RNA sequencing read data, and the partners are searched which one fit the best. All combinations are used. Then this combination is called duplex is formed. Duplex is converted encoded into match, mismatch, deletion, as I have shown there here in this form. Then it is finally converted into matrix form. This matrix is scanned against all existing trained matrix, and then BF3, a machine learning approach, was applied. Okay, and then from that we screened out, classified that if it is going to work or not as a mature microRNA or that. So MyReader this way was implemented, and we could work on different plant system, and found out that it is performing very well actually. So different this slide I'm showing you that different kind of random selection of pairs of this data sets were taken, and for all these pairs you can see the performance was always above 90%. Similarly for human system here, performance was found consistently in the same range. So it was a very reliable software at that time. And across the species also we tested, and it returned very high value of sensitivity, specificity, and accuracy. As you can see here, it was tested across lots of species, and our finding was holding true across all these species. Now we had addressed the problem of precursor finding, we had addressed the problem of mature microRNA finding. Still we were feeling it that something was still missing, and lots of myth we were working actually. So but the biggest myth, or what I can say, because if I say it myth, then since I have been working in this field for a long time, so the old guys who have been working there for a long time, and they still hold it very strongly that no, microRNA formed from these precursors. I usually ask them that if they are coming from precursor, then these precursors are everywhere. So you should have something. We should not get into the traps of precursor and non-precursor. We were realizing that thing, that lots of microRNAs were coming from those regions which were not following this typical precursor form. And if you look into all these things, this minimum fold energy, as I have told you, I have categorized the minimum fold energy into various classes, and you can see here that when this thing was not very discriminating across the minimum fold energy for microRNA and mature microRNA, then there came that this short sequencing platform system. They started telling that there is a certain ratio between 5' and 3' short read pairing, and this ratio must be followed. Even that thing I found that it was not holding correct. So you can see here that the biggest region of that ratio belongs to 0. Actually, it means that one partner is there and another partner information is absolutely not there. So even that theory of them was not working. Then, unlike the user belief, no profound sequence preference exists in microRNA. Also, such discourse does not fill weight with Argonaut sorting theory. One more myth was that it should have U or something like that at the starting or A. Okay, so that's why if a duplex is existing in microRNA, then its the seed region start point 5' region should be the mature microRNA region 5' start point should have U or A. But it was not like that. When we looked into the data, you can see here that lots of different and almost comparable number of representation was by all other nucleotides. So even that theory was not working. Next thing was that they were saying that everything should come from intact stem region. But here I show you that there were certain microRNAs which were coming from the precursor with overlapping nature like this, or here you can see that some of them even crossed into the loop region. So they were constantly challenging. So some of the different examples were there. MicroRNA formation activity is more about combination of other RP than DOSA dicer. We started realizing this thing also because lots of microRNAs were being reported where DOSA dicer implication was not there. So without those enzymes, even they were being formed. More than 50 names of microRNAs are there from coding region, tRNA, tRNA precursor, and huge number of repetitive elements. Okay, they were coming from these regions also, which we used to consider as negative data set. These things were even contributing to your microRNA. Numerous cases of DOSA dicer independent microRNAs, while some microRNAs have been reported coming exclusively from single stranded loops only. Whereas they were not pairing at all, and from this single stranded loops, some microRNAs were coming. Data suggests that more than 60% of a small regulatory RNAs are coming from repetitive elements of genome, and this is the irony because one who studies genome, they simply discard repetitive elements, or they put a clause that if certain microRNA region is being found more than 5 times across the genome, then just drop that candidate. That is such a foolish thing. So people call us scientists, but we have studied with our foolishness also, and we have said lots of such example in past also, but we learn also. But problem is that when you propose new things, you bring this data to very old guys, they become very adamant, their ego gets hurt, and they say, now this is the truth, what we said, that is truth, we don't look into data. Okay, but now they have started realizing their mistakes. So if you go to Mirbase, there also you will find that lots of revision has been done. Mirbase has been criticized a lot for the way they have characterized microRNAs, and lots of changes are now happening. So the last pointer, enough science suggested that we were discarding microRNAs, which were actually microRNAs, but we were considering them as a junk because they were repetitively coming from other locus. Just even if you look into that point logically, many genes, especially if you look into the plant genome, lots of genes are multi-copies, 20 copies, 30 copies of those genes exist in plant system. Okay, and they are studied with repeats. So this then nothing you will find in plant system because they are highly repetitive. So these kinds of erratic things were happening with microRNA biology, and we were realizing that, and then after that we changed our style of working. Just showing distribution of different regulatory small RNA data, how they were coming from different repeat regions, and they were enriched for repeats. Various kind of repetitive families were producing different microRNAs, and these were nothing like that we were telling you without data. They were having sequencing data, and these sequencing data we mapped across the genome, and we found that thing. So finally, why I'm showing you that scientists were celebrating that they developed tool and software to detect precursor microRNA, and they were happy. But Mr. MicroRNA is here hiding behind a wall and laughing on us that you are still far away from catching us, because just it is not like that you find precursor and this kind of a structure and then you define that I exist here, because they were in actually very small component of a large lesion, which I am coming to cover in the next slide. So big question is that keep on searching for conventional hairpin loops like this, identify the regulatory small RNA, because main question is there is small RNA system and how they are coming, different sources are there, they all should be recognized instead of wasting time just recognizing hairpin loops and this and that. So here in 2015 we published a paper in NAR and where we reported this thing, all thing here. So three aphorisms, sutras, there are three sutras that if anything recurs, keeps on coming, reflecting itself in different conditions, then you have to believe. Second sutra, expression and protein abundance. Okay, the expression and protein abundance, that if that particular thing is coming and with good expression, it is coming again and again, and it is found across all those proteins, okay, or associated with all those proteins which are implicated in microRNA targeting, because microRNA targets, that is their function, so they should be associated with those proteins. So if they are caught associated with those proteins, you have no reason to reject them there. Multiple interaction proof, and then I give you the proof that they are having lots of interaction with different involved proteins. So like different interaction proteins, the most important is dicer, because dicer is required to put chop the double stranded RNA, then there is DROSA association, and then risk argonaute complex, which is the most important one, which places this duplex on the target mRNA and takes out the guide sequence and put it for the targeting of the messenger RNA and stop the translation. So anything which is working as microRNA will always be found associated with argonaute data. Okay, so by that time, how I got that much courage, I share you that. That courage I would not be getting had not there been some sort of small RNA sequencing data. That means NGS platform had arrived such a way. In 2013, a paper was published in Cell, 2013 or 14 like that, 13 perhaps. So what they had done, that they had released the atlas of cross-linking of RNA and protein in human system. And that was a big resource actually, and big revolution actually, because there they had introduced hit clip and clip sequencing, where you cross-link a protein with interacting partner, the RNA. So they arrest as it is and rest of the thing are degraded. So you find out that, yeah, this protein was interacted with this RNA. And this data was very recently released. I used that data. So for Argonaut, we got it. Then for risk associated complex, other members like TRBP2, which is important for transferring microRNA from duplex to risk complex. So that delivery agent was also found associated. So we had all this data, and beside that, we had knockdown data. So knockdown, knockout, all this data we were having, where Dyson knockdown were done, then DGCR knockdown was done, and this DOSA knockdown was there. So all these kind of multiple interaction proofs we are having, and 25 cancer conditions data were thankfully given by TCGA, the Cancer Genome Atlas, which is an open database. If you write letter to them, then they allow you access to that data set, and that data set was with us for 25 cancer conditions and corresponding normal conditions. So we were playing with lots of huge amount of big data, you can say, that we were having, and that guts we got from such kind of data only. So this is the general basic workflow, how we worked. Small RNA reads were taken from TCGA database, short read archive and geo database from NCBI. Read mapping was done across the human genome. Unmapped reads were dropped. Count of mapped reads for each condition was measured. Then abundance in each experimental condition was also measured. The small RNA having maximum mapping reads below 5 in any experimental condition were dropped, because below that the credibility issue arises then. Or small RNA reads which were expressing just single condition were also dropped. So we made that very much necessary that they should exist at least in two different experimental conditions, not even in same experimental condition with different samples. No, two entirely different experimental conditions, and minimum 5 reads should match. Then target prediction we did. We had developed our in-house software, Tariff, and other third-party software we took, and from there we got the confidence that if the found small RNA is there, then it should be targeting. So targeting we did, and we found possible targets. Then all these possible targets were scanned across the recently released cross-linking data or against the knockdown knockout system. So Argonaut, CLIP, CLASH, and RNA-seq, microarray, protein abundance, and expression data, all these were screened. And based on all these things, we identified that more than 11,234 regulatory small RNA were existing alone in human system. And while in mid-base, they were reporting just 2,000 or something like that. And actually, there are lots of regulatory small RNA which are actually working, and some sort of symphony is going on there. They different combinations come. If you don't look into kind of conditions, you are never going to know that which part is functional, which part is not functional. One of the major strength for our study was that we considered a large number of these experimental conditions, 25 cancer conditions and corresponding normal tissue conditions. So we were dealing with a good number of different. So even for a normal cell, you must be knowing this. Today in present time, we work on single cell genomics. About single cell genomics, it tells you that even for a given tissue, different cells of that tissue behave very differently. The tissue is same, but cells differ terribly. If you look into some cell at certain distance, they behave differently. Another batch different behavior. So kind of possible experimental conditions or existing conditions in a cell system are almost infinite. Okay. And from that, even the 25 is very small pool, but for data analysis, that was sufficient. And just to satisfy the ego of big daddies here, 11 out of 11,234 RSRNA, we showed them there that actually out of that data, around 3,000 of them were following their traditional canonical model of hairpin loop structure also. So not like that all of them were not following. 3,000 of them were following, and they displayed dosha dependence. So if dosha was not getting expressed or it was hit knocked down, then these regulatory small RNAs were not expressing themselves. So they were typical that sort of regulatory small RNA, which they were calling the typical canonical microRNAs. But beyond that, majority of them were beyond that. Yeah, and I'm writing here 80% of all the RSNA were found associated with TRBP. TRBP is very important protein because it is the protein which is responsible for loading a microRNA into the RISC complex to target and stop the process of translation. So that study we concluded with pre-manifestation. We found that complex repeats rule the genome. So if you are discarding them, you are doing big mistake, because with that you are going to lose lots of regulatory small RNA information. Then several of the regulatory small RNA could demarcate well between cancer and normal states. We were able to do that thing. And one cancer specific thing we found that this RS RNA, regulatory small RNA, which we had identified 981, it was associated with most of the cancer conditions. This one was always coming with cancer condition and going away in the normal corresponding tissues. And these hub systems we identified there, that these were always affected by this microRNA. And adipogenesis and focal adhesion, all these terms were always enriched insulin signaling. So this was a very critical rsRNA, and we experimentally then proved it also. We performed some PCR based qPCR analysis, and we proved all these points for this RS9881. We got experimental validation also. So this way, the myth of microRNA was broken. How microRNA look, that thing we got the picture, and precursor, how they are found, we got the picture. If you have not got the reference sequence, reference genome, how to find microRNA, we gave solution for that also. One more solution we gave is the microRNA targeting, because I told you that if microRNA, if they are existing, their value is just because they are able to target and stop, halt a gene. So how to find target? That was also another important question. Actually, we started our work on microRNA with first on this thing actually, but just for narration, I am putting it at this point. So tariff p-tariff system we developed, and this software is also available on our lab webpage. And from there you can find very accurately the microRNA targets. And here the logic was that simply that microRNA, what it does, it falls on the back of a messenger RNA. Once it falls, it stops, it halts translation. Process of translation is halted and job is done. So in order to stop, it needs to arrive on the back of this target mRNA. For arriving there, it needs open structure or something which allows it to come there. Some openness, some local structure region flexibility is required here. So that situation, I just imagined that it was very similar to plane landing on the runway. The plane does not land if the runway is not clear. So that runway structure we have to derive, and I relied again more on dinucleotide density than the de novo structure discovery. Okay, and here you can see this is very accurate software, and dinucleotide density, when were fed into and trained the support vector machine, they were capable for very accurately classify and characterize the targets if targeting is happening. And you can also see the various patterns are being formed here, and all these patterns are like allowed kind of patterns. So if you cluster these patterns, this pattern could be classified into different clustered actually. So all microRNAs, they follow this kind of pattern of interaction actually, and they belong to one of these clusters. So that was also incorporated, and the very accurate system was developed of tariff and p-tariff. You can see here accuracy, I have shared it, and this is very popular, most popular accurate program up to that time was TAPIR, and it outperformed it. And we have implemented whatever we implement them with multi-core processing capacity. So if you have got a multi-core processor, obviously you get most of the even mobile phones are coming with multi-core processors today. So our system, they utilize those multiple cores, and in parallel it processes, so you have got also very fast processing capability. Now very important thing, here I come. MicroRNA is not parasites and autonomous entities. Why I am writing it like that? Because the big daddies had also said this thing, that microRNAs are parasites, they are not autonomous, they live into the intronic regions of genes, and when the genes are transcribed, then just like a byproduct, they also come out and they are expressed. And now just imagine the irony of these great people. Okay, they call it very important thing. MicroRNA is a very important thing. Okay, same time they say that they are byproduct of a splicing process. Okay, and whenever a gene will be transcribed, bhai sahab unka mood jab hoga, to then microRNA will be released from their cell and gel. Okay, it is not like that. If something is so much important and it is so much spatiotemporal, which is defining your cell system, tissue system, then it is never going to be that much parasite that they depend upon transcription process of their host gene and then they will be happening. So that thing also the challenge. And you would not believe, whenever you speak something new thing or you challenge these guys, then you are always put before some kind of peculiar kind of treatment you get when you try to publish paper. So we had very difficult time proving this thing, and this is going to be a big thing to learn also for you guys here, that we carried this fight for almost 12 years, something like that. For past 9 years, in 2011 we published this paper, and this after 10 years we again published the same paper on the higher and big importance of all these elements. Similar thing we repeated. So we showed here that microRNAs, actually the precursor regions of microRNA, whole set of regions which are enriched for binding for RNA binding proteins and transcription factors. So they have autonomous capacity actually. They are not like parasite that they sit and depend upon the host transcript system. They also exist in the non-genic region also, outside intronic zone boundaries. How they are transcribing there? That means if they are transcribing from there, then they must be having their own machinery. So that we found here, and we reported that the microRNAs actually, they are independent of host, and they have their own autonomous system and which they use, and transcription of their own happens, and then processing by RNA binding proteins happen. At that time, data for RNA binding proteins were not there because in 2011, every sense of regulation was associated with this transcription factor. That only regulator is transcription factor, so every regulation will happen at transcription. And you are studying a post-transcriptional regulator like microRNA with this theory. So full of RNAs was happening. So we reported that RNA binding proteins data was very much scarce. Somehow we managed to prove that, and as you can see in the plot here, that here is the mature microRNA region, and you can see that it is studded with lots of transcription factor binding sites and RNA binding sites, and they were actually also forming composite regulatory modules also, that when this combination will happen, then transcription will happen, another combination comes, then transcription will happen. So lots of these kind of things were. We were able to prove it through using different kind of expression data, mostly based on microarrays. At that time, sequencing data access we didn't have. So we used microarray expression data, and based on that. But we could not move beyond that because we had lots of limitation, data were limited, and with that much scarce situation, we were able to tell this truth to the world. We reported that thing, and then we arrived to our latest part. MicroRNA formation is much beyond the traditional tale of dosa, disa, tango, because I had reason to say so. Here you can see that lots of RNA binding proteins were involved. So obviously microRNA formation was not just a matter of dosa dicing, chopping it. RNA binding proteins other than that were found associated by us. And in the meantime, this ClipSec data, about which I told you that in Cell a paper appeared, and that paper had published lots of interaction, cross-linking experiment was possible. Finding the partner, interacting partners was possible. So that data was there. Now you can see 2020, now we are in 2021, also 2022. This data has shot up. So ClipSec data, combined data, if you do different techniques, here different techniques are quoted. And if you combine, then this blue plot is showing that collectively how much data is there. So lots of RNA binding proteins are actually binding this precursor, processing all these things, and duplexes being formed. And that we were able to get from ClipSec data. And what RNA binding proteins are? You see here. They are regulators of regulators, actually. So almost every process performed by RNA, or every RNA, is actually controlled by RNA binding protein. So any process you take, no RNA can survive without RNA binding protein. It needs association and it needs RNA binding protein for its function. So microRNAs can't be an exception, and that was proved here. DOSA dicer are uniformly expressed enzymes. So DOSA dicer enzymes, they are almost everywhere. Wherever you find double-stranded RNA, this man dicer guy, that boy will come and cut it, chop it, and produce a duplex. Okay. So it is getting, this data is from various tissues of human cell system. You can see that DOSA dicer, both here, both are being expressed almost consistently in all these tissue types. So if they are being expressed in every condition, okay, then why all the microRNAs are not getting expressed in every condition? Because microRNAs are just byproduct of DOSA dicer activity. So whenever DOSA dicer arrive, they should chop all the existing microRNA, and every microRNA should express themselves with equal amount of expression, but it is not happening. MicroRNA are highly spatiotemporal. For any given condition, a certain set of microRNA you will find in cell, no other microRNA will found. Even you will find their precursor in the cell system and mature microRNA not forming. Such kind of high level of regulation is happening with microRNAs. How one can ask what just tell that DOSA dicer are the main thing due to which microRNAs are formed? No, it is not possible. Something which is getting expressed all the time, and there is something which are highly spatiotemporal, very choosy in their expression. So this relationship is not like that. And then we moved into our further work. So we had thankfully now lots of data by this time. 10 years back, we didn't have data. But now we had lots of data. Data of which form? We had protein-protein interaction network data. We had lots of information. At that time, hardly you had information for 100 or 50, something like very small amount of RNA binding protein. And even you were knowing them as RNA binding protein. Even their data was not there. But today, there is proof that there are more than 3,000 RNA binding proteins. Even those enzymes which were once found in TCA cycle, Krebs cycle, or glycolysis, they are now being annotated as RNA binding proteins. It is such kind of transformation. Those types of transcription factors, which were earlier recognized as transcription factors, those types of RNA transcription factors are now being called as RNA binding proteins. They are found binding to the RNAs. So RNA binding protein data we were having through ClipSec and tractome capture like things. Then precursor microRNA data through RNA-seq we got, and mature microRNA data through small RNA sequencing we got. And using all these things, we developed Bayesian network system. And Bayesian network system was used to derive how the microRNA, different steps of microRNA processing was happening. So from primary to precursor microRNA formation, then precursor to mature microRNA formation. We developed model for almost 1200 mature microRNAs in human system, and for the first time we have revealed that these microRNAs are way beyond DOSA dicer. So I'm just showing you how for different RNA binding proteins we got data of ClipSec sequencing. We consider RNA-seq data, small RNA-seq data, and ClipSec data for common conditions so that we could build a complete model. And for 32 different experimental conditions, we could derive this data. And you can see that lots of them also show the preferential binding across RNA binding proteins. Like here, you can see for some, and just for a few, I'm showing it here. This is the basic set of steps involved in doing that thing. So expression data are used. Based on that, there is something called structural learning, which derives the reasoning that how, in which direction this interaction is going on, which kind of steps are involved. So in back chain protein-protein interactions, then that particular protein-protein interaction results into what set of new interactions, and how finally all of them deposit upon the back of this precursor microRNA system, and from that precursor microRNA system, this particular most possible interactions are derived here. The microRNA biogenesis models are the final output we got. So we also validated that how much accurate our findings was there. So the kind of relations which we derived using Bayesian network, we found them significantly specific and very much holding correct when we compare to the randomized data. And you can see here that in randomized data, the proportion was much lesser for random combination. And this, here you can see that these were holding with high significance. And these are the number of experimental conditions. Like here is a tissue number, that how many of these combinations were shown, like just in one experimental condition, 19.2% combinations were there. Then in two different experimental conditions, like two different tissues, 19.5 were here. And just like that, like you can see here that 49% were having getting associated for more than four more than four experimental. Total 8 tissues we had considered, different kind of tissues, and they were holding themselves correct across all this. Finally, we found AR2, one of the proteins which was highly associated with microRNA formation. First time we were reporting. There are other proteins also, lots of protein are there, but we focused upon AR2 because it was associated in precursor, primary to precursor formation also, and from precursor to mature microRNA formation also, and one of the most abundant. Okay, so we performed knockdown study also, experiment on AR2. So we found that if the dicer is knocked down, so then still this one remains expressive. The microRNA is still getting formed. But if AR2 is knocked down, okay, then this particular microRNA formation is not being done. So lots of these things are there. We even proved it experimentally that yes, dicer independent system is there, and microRNAs are dependent on other RNA binding proteins. Lots of relationship also be derived that were cooperative or mutually exclusive or competitive. That competitive means that if this RNA binding protein comes, then this will be not able to act. So it was like that. There lots of such combinations and relationship between various RNA binding proteins were also derived, and we presented them. Then we found that these microRNAs, actually lots of work have been done them that microRNA can work as precursor, biomarkers. So biomarkers like that they can distinguish between disease state or normal state of different kind of conditions of a cell system. So we were just curious that if they are holding so much correct, if microRNA formation is associated with RNA binding protein, then those RNA binding proteins should also mimic similar kind of separation which we attend through their associated microRNA. So we try to distinguish between different normal and cancer conditions, and microRNAs, what kind of separation we got from microRNA, the similar kind of separation we got if we replace those microRNAs with their associated RNA binding proteins, and similar kind of classification between disease and normal condition we got. So they were reflecting that there also. So finally, here we arrive that if something is there, and if RNA binding proteins are deciding the formation of mature microRNA formation, and in such a spatio-temporal manner, then why not make a software which will stop the need of doing microRNA profiling experiment at all itself? And with that kind of idea, we started working on this software, and we implemented this Bayesian network system. And what we had found that all the association, all these paths detected by Bayesian network system were vectorized and used as a feature actually. So you see, this entire finding was automatically transformed into feature selection method, and they become feature extractor actually. The Bayesian network became the feature extractor. And these features were, okay, fed into this XGBoost regression learning, and using that, we were now able to just predict the microRNA very accurately. MicroRNA profile, what kind of microRNA they are going to be in the system, and how much they will express without doing any experiment on them. You just need to provide me RNA-seq data, general RNA-seq data, so you don't need to do any separate microRNA sequencing data profiling or microarray, anything like for microRNAs. You don't need, you just do one thing for RNA-seq data for general cell system. That is very common thing. You just feed to this system and it will tell you that what kind of microRNA are getting expressed. So you get the plot here, and plot I am showing the one with actual what expression was predicted and what we predicted. So this entire software system we have implemented on our website. You can put your data here in sample gene ID for human. You provide your expression, raw expression data for RNA-seq, and from there it will build all the thing. Accordingly, it will fit and predict the expression profile. From expression profile, you will also get bubble chart representing various kind of highly interactive it is. From there, you will get all these things and details. You click on select any microRNA, it will show you different targets, how it is enriched. Then you can go to pathway, in which pathways are getting affected. Everything you get there in the result page. I'm showing you the case of 6 such microRNAs. They were tested across different kind of experimental samples, how their expression was predicted in for all these samples, and how close accurate it was to the actual. So you can see here most of them were around 90% accuracy. Lowest accuracy was found 87%, and highest accuracy was for 94%. Average accuracy was 91%. So future is deep reaching. I conclude my presentation here, and most of the software which I showed you, they were with machine learning. Now we are shifting to deep learning. Deep learning uses machine intelligence, okay, and those features which you need to derive, okay, that are fed into machine learning system. But in deep learning system, even those features which you can't detect, actually you can't derive, they are actually derived by deep learning. So we have almost done 2-3 such kind of software here, and you will be, if you visit, you will find few of them there. And for microRNA system also we are implementing, and first thing which is about to come is MIR-High. Call it MIR hybrid, microRNA hybrid system, or Hydra you can also say. So here we have actually incorporated deep and shallow learning. So with this, I conclude my lecture here. I had thought of showing you some hands-on also of the software which we had developed here. But if you visit to this website here, scbb-bt.resin, you will get our all software there. Some of the web servers are now not working, but we have provided a standalone version of those programs you can download on your machine, and you can run and get the result there. We have maintained all them on GitHub also. So from there also you can download. And for MIR-Biome, this here, if server is working, you can put your data here, and on web server you can do predictions sort of things also. Even this is also downloadable. We prefer to download things. Okay. With that I conclude. I just request that from next time, if possible, then use some other interface or interaction so that it could support Ubuntu Linux system or any Linux system, because most of the scientific programming are done on this Linux or OpenBSD or BSD Unix system. So I'm sorry that I would not be able to show you any hands-on training here, just because I'm working on Windows system. Okay, thank you. Any question? You're most welcome.
So I have a comment. Dr. Ravi Shankar, congratulate you.
Hello.
Yeah, this is Mitali here. Congratulate you for
mic
your work.
work.
You're not
Hello.
able to hear. Hello, can you
Hello.
hear? Can you hear? Can you hear
I'm
me?
unable to get any voice. Yes,
Okay.
conclude it. Thank you very much. Stop
Okay.
sharing कहाँ से?
Okay. Is
एक से
he
इसका
able to
है,
hear?
एक
He
से
spoke. I'll talk to him later. He's done some very
Dr. Ravi had some issue with the speaker, so I will then request the participants and even, in fact, panelists to
ठीक है. Bye bye.
write in the chat box.
Hello. Are you on?
प्लीज वेट सर, वो एज मैसेज आया हुआ चैट में पूछ सकते हैं ना.
Hello. Or we tried, but my voice is my voice was coming to you or not? But your voice is not coming my side. Yeah, that we have, that is working actually. That is working, but we inbuilt speaker ऐसा है ना?
इसमें से इनबिल्ट नहीं है. एक सेकंड, एक सेकंड.
हेलो. बोल बोल.
बोलो.
आवाज आ रही है? नहीं नहीं नहीं. Yeah. That somebody has just typed that. I'm just replying on chat box only. Okay.
डायरेक्ट लिखा हुआ था गो टू, और फिर वो इसी के बीच में टाइप हो गया.
Okay, thank you so much, sir, once again. It was a great pleasure to have you here. So we would like to now close for the today's session, first half session. So in the second half, we will have a third session.
Voice, पंकज जी, voice is coming now. I can listen. So कोई पूछना चाहे क्वेश्चन तो मेरे से पूछ भी सकते हैं अभी और फर्दर कहीं पे
Okay,
टाइप कर
okay,
लीजिए
great,
पे
great. Ultimately, it worked.
लास्ट जब खत्म हो रहा है, लेकिन ये है कि आगे से सेशन जो है ना थोड़ा सा हो सके तो इस पे हम लोग जूम पे या नहीं तो इस पे भी कर सकते हैं। क्या नाम है
Sure,
उसका,
sure. We will
टीम पे,
take care of
MS
this.
टीम पे, है ना?
Yeah, we will take care of this. If any participant would like to interact with Dr. Avi Shankar, please raise your hand so we can promote you to the panelist.
ठीक है, किसी का है क्वेश्चन तो ठीक है, फिर
Yeah, I
अदरवाइज
think Ms. Pooja Parmar wants to talk. So
Yeah.
please unmute yourself and you can ask.
Very good afternoon, sir. Sir, thank you
Good
very much
afternoon.
for very informative session. In fact, I was not aware that this much change has occurred in study of this mRNA. So, sir, indeed, it was very informative lecture. Sir, I have one query
Thank you.
regarding
Yeah,
the
please.
software developed by you. Sir, I have
Yeah.
many RNA-seq data for different expression levels of rice genome. So, sir, can this can I submit that in the software developed by you, or is it for specific?
For which purpose? Which purpose? Because there are
Sir,
many
for
purposes.
finding miRNA, like as you
Yeah, for
told.
finding, yeah, for finding microRNA you can, because
Yes, sir. So it is
for
rice sequencing data. So that is why
yeah,
I'm.
you can provide no issue.
Okay,
You have
okay.
RNA-seq data, na?
Yes,
RNA-seq
sir.
data, na?
Yes, yes, sir. Yes, sir.
So what I will do, that we have not released that software so far. MirHi can do that part.
Okay, sir.
Actually, we had even benchmarked MirHi across rice genome itself.
Yes,
So
sir.
we
Yes,
are
sir.
actually having most potential microRNA precursor region for rice also. So
Yes,
what
sir.
you can do, that you can simply look into those regions. We will be providing, you just write me a mail on Gmail or my HBT ID. We will
Yes,
be
sir.
providing you access to those files. From
Yes.
there, you can find out your data, how your data is mapping or something useful is there or not. We have already run across the rice genome, so why to redo that thing? We have already that thing.
Okay, sir.
Yeah.
Thank you. Thank you so much.
Yeah. Anybody else? और किसी को पूछना है क्वेश्चन? पंकज जी, I think then it is done. I conclude the session here, and thanks to all of you for having patience to listen to such long lecture, and that too without any hands-on session. I'm sorry for that part.
No worries. So thanks for your time, sir, and it was really good. Thanks, and let us close the session of the first half today. So I was just saying that we will have third session today by Dr. Dilpreet Kaur. Very exciting session on matrix methods in data science, starting at 3 p.m. So I will request all the participants to join on time at 3 p.m. so that we can start in time. So thanks again, sir. So we will be closing now.
Good afternoon, everyone. It is a great pleasure for me to introduce our speaker, Dr. Dilpreet Kaur, for today's talk. Dr. Dilpreet Kaur completed her BSc from Punjab University in 2007 and her MSc in Mathematics from Punjab University in 2009. She also completed her PhD from the Department of Mathematics at the Indian Institute of Science Education and Research, IISER Mohali. She was a postdoctoral fellow in mathematics at IISER Pune. Currently, she is an assistant professor, Department of Mathematics at IIT Jodhpur. Without any further delay, please join me in welcoming Dr. Dilpreet Kaur to address the session on matrix method in data science. Thank you, ma'am, for coming. Over to you.
Thank you very much, Monica, for a nice introduction. Let me share my slides first. Okay. I hope you can see my slides, right?
Yes, ma'am.
Okay. So, can I see participants in this call? So, today I will be speaking on matrix methods in data science. This talk will be very basic. I will not be assuming anything except that people know how to multiply two numbers or how to add two numbers. So it will be very basic, and I think that everyone will be able to follow, rather find it too easy to follow. Okay, so even then, if you find that you are not getting something or I'm going too fast, please stop me. I would like to repeat that thing, and I want everybody be with me during this whole time. Okay. So first we see what is a matrix. A matrix is nothing but an array of numbers arranged in rows and columns. So this is a M cross N matrix. By M means we have M number of rows. So this is first row, second row, continuing so on. M is some integer and we have M number of rows. And by this N, the second factor in M cross N matrix, N is number of columns. So here we have N columns. This is first column, this is second column, this is third column, this is nth column. Whenever we are writing an arbitrary matrix, a entry like here, okay. So suppose we are talking about this entry. This is written by A23. So the first subscript is row number. So 2 tells me that it is in second row. The second subscript is column number. So 3 tells me it is in third column. So that is how we write matrices. So it is also, in short, it is also written as Aij, where this is this denotes ith jth entry. Means ith jth entry. By ith jth entry, I mean the entry in ith row and jth column in M cross N matrix. That means it has M rows and N columns. I hope the definition of matrix is clear to everyone. So now there is a question for students. So what do you see in this picture? I hope people can write in chat and answer. What do you see in this picture? I'm sure like most of you are saying that, okay, you are seeing a flower, in background we can see leaves and so on. But how are you able to answer this? Thank you, Priya, for answering. So what goes in your mind when you identify picture? So basically, we don't understand that what goes in our mind when we identify this picture. With years of training, we automatically identify a picture. If we know someone, and we see that person's picture and we say, okay, this is his or her picture. But if I ask you to write a code for computer to do the same job, that if I feed this picture in computer, then computer should be able to tell that this is a picture of flower. That is not easy, and that is very active area of research in machine learning. Okay, So to understand that how computer will identify a picture, we need to understand that how does computer stores a picture. So this is a small picture of letter 8. So I have in particular taken a picture which does not have any very good resolution. So you can see these small boxes in this picture, right? These boxes are called pixels, and the dimension of this picture is 24 cross 16, which means that it has 24 pixels around the length, this height, and 16 pixel across the width. Okay, so this is 24 cross 16 picture. It has 24 pixels around the height and 16 pixel along the width. Okay, now how does computer will store this picture? Okay, the computer stores this picture in the form of numbers. So all the every pixel will have a pixel value which will range between 0 to 255. This is a grayscale picture. So first we are discussing that how does computer store a grayscale picture. Okay, so 0 is for black, the pure black color, and 255 is for white color. So any value between which is close to 0 is more dark pixel, and the value when it is close to 255, it is the light colored pixel near to white. Okay, so computer stores every picture in form of a matrix. So this is now a matrix of 24 rows, because along the height I have 24 pixels, and 16 columns because along the width I have 16 pixels. So every image in computer is stored in terms of a matrix. And in case of image processing, these matrices are called channel. Okay, so if you have a grayscale picture, it has one channel associated with it, which is basically height number, which is basically m cross n matrix, where m is number of pixels along the height and n is number of pixels along the width, and the value, each value varies between 0 to 255. This is how a computer stores a grayscale picture. So let's see it more closely in this slide. So this is how the previous picture will be stored in computer. So here this is a 24 cross 16 matrix. The matrix entries are varying between 0 to 255. This is how the computer will store this picture. Okay, I hope this is clear to you, right? Is this fine? I stop here for a minute to understand if this is fine with you. Please reply. Because I'm going to ask one question. Thanks, Priya, for answering. So you have a computer, hope you have a computer with very good resolution. So the standard HD resolution of a computer is usually 1366 into 768. What does this mean by resolution of a computer? That means if I'm seeing one picture on my computer, if picture has good resolution, then it will have 1366 pixel along the height and 768 pixels along the width. That is the meaning of resolution of a computer. So that makes, multiplying these two numbers, that makes a picture will have these many pixels, 1049,088. Now, after you understand that how computer stores a grayscale picture, and you understand what do we mean by resolution of a computer, can you tell me that how many grayscale images we can see on my computer screen, suppose? How many grayscale images I can see on my computer in my lifetime? So I can read any book on my computer, any book which is written till date, or any book which will be written in future. I can read all the books. I can see the picture of any bird, any animal, any dinosaur, or anything on my computer. So what do you think that how many grayscale pictures I can see on my computer? Any idea? Please reply. What do you think, that is there any limit, or I can see unlimited images on my computer? What do you think about it? Is anybody replying, Monica? This is very interesting.
I'm not able to see like anybody replying to it.
Okay. Okay, unlimited Priya is saying. Okay, that is the answer I wanted to get. But now let's see, what do we mean by unlimited? In language of mathematics, we say that unlimited means infinitely many. Okay, so now you see, I explain, actually you can't see infinitely many images on your computer. Here I try to explain why. Suppose I have this grid. Okay, suppose this is a picture and it has 4 pixels, 1, 2, 3, and 4. And suppose I have only 2 colors to give to 1 pixel. So suppose I either can keep it white or I can paint it black. I have only 2 colors. Then how many these 4 pixel pictures I can have with 2 colors? See, for the first grid, for the first pixel, I have 2 choices. Either I paint it black or I keep it white. So I have 2 choices for first pixel, and I have 2 choices for second pixel as well. And I have 2 choices for third pixel, and I have 2 choices for the fourth pixel. So total number of pictures to get from this 4 pixel set with 2 colors is 2 to the power 4. What is 2 here? Here 2 is number of colors available to us, and this 4 is number of pixels. So the total number of pictures you can see on your computer, grayscale pixels we are talking about, is 256, because we have colors available from 0, 1, 2 till 255. So I have total 256 numbers to the power number of pixels. Okay, so on your computer you can see at most, okay, on your computer you can see at most these many pictures. Okay, and how large this number is? This number is so large that if you print it with normal font on a paper, it will take 80 pages to print this number. Here we are writing 256 to the power this whole number. So it will, if you solve this, and it will contain so many digits that it will take around 80 pages to print this number. So this is a very large number, but still a finite number. And this covers all the possible books in the world, because usually books are black and white. And all the possible grayscale images you can see on the computer. So this is a very, very large number, but still a finite number. So because of this, the way computer stores the picture, and because of the limit on resolution of a computer, the number of pictures you can see on your computer is not unlimited, is not infinite. This is a finite number, but it is a very, very large number. Okay, is this fine? Okay, so here we learned that how computer stores a grayscale image. Next, we understand that how does computer store a colored image. So this is, suppose that every color can be generated from 3 primary colors, red, green, and blue. If you know a painter, the painter will tell you the 3 primary colors are red, yellow, and blue. But in computer, we take red, green, and blue. So a grayscale image is composition of 3 channels. One channel is corresponding to red color, other channel, here these channels are formed. Other channel is formed by green color, and the last channel is formed from the blue color. Okay, so here again, this will be a channel will be a matrix, M cross N matrix, where M is number of the pixels around the height, and N will be the number of pixel along the width. And each pixel can take value between 0 to 255, where 255 will be the dark red color, etc. And a color image is composed of 3 channels. Okay, so this is how computer stores a colored image. Okay, so this was my first motivation to learn matrices. Why do we learn matrices? Because it is some matrix with which computer stores an image. Okay, so whenever computer stores an image, then it is actually storing a matrix. If it is storing a colored image, it is storing 3 matrices for that picture. If it is storing just a grayscale image, then it is just storing 1 matrix corresponding to that picture. So this is the one motivation to learn matrix. The other motivation to learn matrix is system of linear equations. Okay, so we come across system of linear equations many times during our day, and we solve them without our knowledge. So what is the system of linear equations? It is just like this system. This is a system containing 2 equations with 2 variables, x and y. The unknowns are called variables. This is system of a linear equations. What do we mean by solution of a system of linear equations? That means we want to find a value of x and y which simultaneously solve both of these equations. Okay, and usually as a kid we have learned 2 methods to solve the system of linear equations. One is graphical method. What do we mean by graphical method? That is, I draw these 2 equations. So this is equation in 2 variables. So when I draw it, I'll get 1 line. This is again equation in 2 variables. I'll get 2 lines from here. I draw those 2 lines, and then I study that graph. That graph will give me solution of these 2 linear equations. Okay. And the second method, which is more popular, is elimination and substitution method. You operate these 2 equations in such a way that you first eliminate one variable and you get the value of another variable. Then you use the value of another variable to get the value of the previous variable. Okay. So let's see how do we use these 2 methods to solve the system of linear equations. So here on my left hand side, I'm explaining the graphical method. This green line is line of first equation, x minus y is equal to 1, and red line is equation of second, line is plot of second equation, x plus y is equal to 3. Now you see this line are meeting at this point. Okay, they are meeting at this point. So this point satisfies both of these equations. What is this point? Here it is taking value 2 and 1. So 2, 1 is solution of this system of linear equations. And on right hand side, I'm explaining elimination and substitution method. So these are 2 equations. I want to eliminate one variable. So if I add these 2 equations, y will get eliminated. If I add these 2 equations, you will get 2x minus y plus y is equal to 0 and 4. So I'm getting 2x is equal to 4. Then because 2 is a non-zero number, I can divide this equation by 2, which implies x is equal to 2. Now I put the value of x in any equation. You can either put in first equation or you can put in second equation. So suppose we are putting the value of 2 in second equation, then we are getting this is equal to 2 plus y is equal to 3. So you subtract minus 2 from both the sides, we are getting y is equal to 1. So from here also we got that system of this linear equation is 2, 1. Okay, these are 2 widely popular method which we also learn as a high school kid in high school. Okay, so this was an easy problem. we had only 2 equations, we had only 2 variables. But let's complicate this problem a little bit. Now we have these 3 equations and 3 variables. So now which method you will like to use? We already know 2 methods, graphical method and elimination and substitution method. In 3 variables, this will form a plane. And in 2D, it's difficult to draw, at least for me, to draw a plane. So I will show you elimination and substitution method. Why I want to solve it again? Because I want to make a point regarding number of steps involved. So bear with me, let's solve this together. We have these 3 equations. So suppose we are adding 1 and 3. What we are getting? 2x minus y plus y is 0. So we are getting 2x is equal to 4, which implies x is equal to 2. Now suppose I'm adding 2 and 3. So I'm getting 2x plus 2y plus z minus z is 0 is equal to 6. This dividing by 2, I'm getting x plus y is equal to 3. Put x is equal to 2 here, I'm getting y is equal to 1. Now you put x is equal to 2 and y is equal to 1 in one of these equations, then what I'm getting? So we are getting 2 minus 1 plus z is equal to 2, which implies z is equal to 1. Okay, so solution of this system of linear equation is 2, 1, 1. Why we are solving this system of linear equations? So let's count how many steps we did here. So adding these 2 equations were 1 step. From this first step, we got value of 1 variable. In the second step, we substitute this value and get the value of second variable. So we did only 2 steps here. When we increase the number of variables as well as number of equations, then see how many steps we are doing. Here we add these 2. This was actually easier. So we did 1 step. We add these 2. We did second step. Then we put value of x in this equation. That was the third step. Then we put value of these 2 equations in one and get the value of z. That was the fourth step. So from this method, you can clearly see that as the number of equations and number of variables will increase, the number of steps required to solve the system of linear equations with elimination and substitution method will increase, and they will increase really rapidly. Okay, so here I am explaining that, how can 2 planes intersection of 2 planes, okay. See, here when we are solving this system of linear equations, this is equation involving 3 variables. So that means this is the equation of a plane. So these 3, if you plot these 3 planes on same coordinate set, you will be lead into one of these situation. All the 3 planes can be parallel to each other. In this case, they are not intersecting with each other. Either it can be this situation where all the 3 planes are intersecting on a single line. Then there is this situation that there is one plane and 2 planes are intersecting the third plane, but at different lines. Then we have all the 3 planes are intersecting each other, but they are intersecting at different lines. These 2 are intersecting at this line. This green and this yellow green is intersecting at this line. Brown and green is intersecting at this line. And here these 3 planes are actually intersecting at a common point in middle of it. Okay, so these are the ways in which system of equations can behave. If we plot 3 equations with 3 variables, you may get one of this situation. So how does these graphs are related with system or solution of system of linear equations? So let's come into this. So if plot is like this, that means they are not intersecting at all. In this case, system of linear equation has no solution. Here they are intersecting at entire line, so we will be having infinitely many solutions. Here we don't have any solution, we have no solution. Here we have unique solution. So, but okay, I'll come to this later on. So now from these, this is a graphical method for 3 variables. This is elimination method for 3 variables. From these two set of system of linear equations, we learned that the complexity to solve a system of linear equation will increase with increase of number of variables. But if you are dealing with a data in real world, that could have thousands of variables. So if you are using one of these methods to solve that system of linear equations, these are the limitations of these methods. So graphical method is applicable only when number of variables is not more than 3. So if number of variable is more than 3, then you can't imagine the graph of that system of linear equations, because we live in three-dimensional world. So for any of us, it is very difficult and sometimes it's impossible to visualize a system which is having more than 3 variables. And what is the problem with elimination and substitution method? If n is very large and system of linear equation has n variables, then it will roughly take 1/3 into n cube steps to reach at a solution. So when the number of variables are increasing, the number of steps are increasing even faster, because for n variables, we are having 1/3 into n cube steps to reach at the solution. And that will happen if the solution exists. I'll come in a minute. What do I mean by if the solution exists? So that means that it may be possible to have a system of linear equation which does not have any solution. So for example, here I have plotted a system of linear equations with 2 variables, 2 equations, which is not having any solution. So here in graphical methods, so suppose this is basically line x minus y is equal to 0, x is equal to y line, and this line, say if this is 1, this is 2, you can say this is line of x minus y is equal to 2. These two are parallel lines, they are not going to intersect at any point. So there does not exist a value of x, y which satisfies both these equations simultaneously. Okay, so here we are looking at this by graphical method. If you subtract these two equations, you will get 0 is equal to -2. So by elimination method, which is not true. So by elimination method, you can also get that this system of linear equation has no solution. And the other question is, if a system of linear equation has a solution, it may not be unique always. So here is plot of the other part, other extreme. So here, if you can see, I don't know if you can see properly. Here I'm plotting 2 lines. One is with large width, red line, and other one is with less width, is a black line. Both the lines are coinciding with each other. So any point on this line is a solution of this system of linear equations. So this way, this system of linear equation has infinitely many solutions. So for example, if this is, say, both 1, so this system of linear equation can be written as x plus y is equal to 1, and the other equation can be this. Now you see that any value that y is equal to x minus 1 is a solution of this system of linear equation. So if x is equal to 0, no, sorry, this is 0, 1, this is 1, 0, this is 0, 1. So this is x minus y is equal to 1. Okay, if x is equal to 0, y is equal to 1. If y is equal to 0, x is equal to 1. So any number of this type, basically x, x plus 1, any tuple, is a solution of this system of linear equation. Okay. So we have solution 0, 1, we have solution 1, 0, we have solution 1, 2, and so on. No, sorry, I'm doing some mistake. Here 0 and 1. Minus y is equal to x plus 1. No, I'm doing some mistake somewhere. Wait a minute, please. Suppose this is a line passing through. This number is 1, 0. This is 0, 1. Okay. Okay. Then x is 0. It's satisfying 0, 1, 0, 1. Yeah, this is line equation of this line. Okay. So the other equation will be multiple of these because they both are same line. So this system of linear equation will have solution x and x minus 1. So if you are taking x is equal to 1, 0. If you are taking x is equal to 0, then it is coming 0 minus 1. So this should be this line, not this line. Okay. Okay, if we forget this line, then even this is a system of linear equation having infinitely many solutions. This is not line of this system. Okay, this has this solution because it satisfies this solution satisfies this line. Okay. So here I'm trying to answer two questions. One is that there can be a system of linear equation which has no solution, and it is possible to have system of linear equation with infinitely many solutions. So basically when you have system of linear equations which have no solution, the situation in graphical method will be, if it is two variables, then they will be two parallel lines. If it is two variables with infinitely many solutions, then that will be the coinciding lines. And in case of three variables, so this is the situation with no solution because these planes are not intersecting at all. This is a solution with infinitely many solutions because all the three planes are intersecting at this full line in between, and any point on this line is a solution of this system of linear equation. This has no solution because all the three planes are not intersecting at any point. Here also the situation is of no solution because all the 3 points are not intersecting at any line. And here the situation is of a unique solution because these three planes are intersecting at unique point. Okay, so is this fine till here? So till now we have discussed two methods to solve system of linear equations. One is graphical method, another one is elimination and substitution method. The limitations of these both methods are very difficult to operate if number of variables are too many. Okay, so basically we want to say that this system of these two methods to solve system of linear equations are not efficient enough if number of variables are large. So we need some other efficient method, and that method again comes from the matrices. So for that, we need to learn more than the definition of matrix to understand. Okay, so here I'm showing you that how we can add two matrices. So here I have this M cross N matrix. Okay, and here is another M cross N matrix. You can add two matrices if and only if they are of same order. This is called order of matrix. If they are of same order, only then you can add these two matrices. So the addition is entry wise. So this first entry of first column will be added to first entry of first column of second matrix, and that sum will become first entry of first column in the resultant matrix. So we are adding this coefficient wise or the coordinate wise. So this entry will get added to this entry and will get here. So we are adding it coordinate wise. Okay. And so because we are adding it coordinate wise, so we can add two matrices if and only if their order is same, and the resultant matrix will also be M cross N matrix, will be of same order. And addition of matrices has this operation that you can keep them in any order and add them, the result will be same. So this is called commutative. Addition is commutative. So whether I keep A first and add it to B, or I keep B first and add it to A, the answer will be same. Okay. And the second operation with matrices is scalar multiplication. So when I'm talking about matrices, the entries of these matrices are real numbers. So I can multiply this matrix with any other real number. So here this symbol in mathematics says belongs to. And when we write R with double line, that means real number, set of real numbers. Okay. So I can multiply any real number with any matrix. So this is matrix of M cross N order. Each entry of matrix will be multiplied by that real number. So here this is a Greek letter lambda, which is representing any random real number. So when you are taking when you are multiplying a matrix with a real number, in that case real number is given a fancy name called scalar, and it is called scalar multiplication. Okay, so this is, we are multiplying each entry of this matrix with lambda, with that is scalar multiplication of matrix. So we have now two operation on set of matrices. One is addition, which is coordinate wise, and other one is scalar multiplication, where again we are multiplying each entry of matrix with that scalar, and these two operations are compatible. What do I mean by compatible? That means that if I have two matrices A and B and I add them, then I multiply the answer with lambda. That will be same as if I multiply matrix A with lambda first and matrix B with lambda and then add them. In both of the cases, the answer will be same. So that's how these addition and scalar multiplications are compatible with each other. Okay, and there are other compatibilities too. These two real numbers, if you add them and then multiply to matrix, that will be same that if you multiply lambda with matrix first and then mu with matrix A and then you add them, that will also be a same thing. And it's very easy to verify if you have understood the scalar multiplication and addition of two matrices. Next, we come to the very important operation of matrices, that is matrix multiplication. Here you need to pay attention that if A is M cross N matrix and B is N cross L matrix, then their product is M cross L matrix. Now, if I want to multiply two matrices A and B, okay, this is M cross N matrix and this is N cross L matrix. I can multiply two matrices only if number of columns in first matrix is same as number of rows in second matrix. Otherwise, you can't multiply the two matrices. The resultant matrix will be of order m cross l, where m is number of rows in first matrix and l is number of columns in second matrix. Okay. Here is how we multiply two matrices. So this one has two rows. This is 2 cross 3 matrix. This one has 1, 2, 3 rows. This is 3 cross 2 matrix. Here number of columns in first matrix is same as number of rows in second matrix. So we can multiply these two matrices and resultant matrix will be 2 cross 2 matrix. And how do we multiply them? So here the first entry of this resultant matrix is coming from first column, first row of first matrix and first column of second matrix. And we multiply this first row with first entry of first row with first entry of this column, second entry of row with second entry of column, third entry of row with third entry of column, and we add each one of them. Okay. And similarly, to get this second entry, what we do, we'll take first column from here and first row from here and second column from here. So 1 into 2 plus 2 into 4 plus 3 into 6. That will give us 2 plus 8 plus 18, which is 28. To get this entry, you will take second. So this is what? Second one entry. Second row and first column. Okay. 1, 4 into 1 plus 5 into 3 plus 6 into 5 will give you this. And the last, this is second 2 entry, second row and second column, you will multiply. Okay, this is how we multiply matrices. Is it fine? Have you seen matrix multiplication earlier? Okay, Priya is saying yes, good. So have you ever thought that why matrix multiplication is not coordinate wise, like addition is coordinate wise? So why don't we multiply matrices coordinate wise? Anybody? To answer this question that why matrix multiplication is not coordinate wise, I'll give you two examples. One is how Facebook uses matrix multiplication to maintain our friend circle, and other one is coming in next slide. So suppose there are 5 people who are using Facebook, and if they are, so we are plotting a graph between them. So I am drawing the edge between 1 and 2 if 1 is friend of 2. Okay, if 1 is friend of 2, then definitely 2 is friend of 1. And so here this graph says that here is network of just 5 people. 1 is friend of 2, 1 is friend of 4, 2 is friend of 4, and 2 is friend of 3 as well. 3 is friend with 2 and 5. 5 is friend only with 3. Okay. So now, so you must have seen suggestions from Facebook that you may know these people, etc. How Facebook gives those suggestions. So these matrices plays an important role there. So here we are writing adjacency matrix of this graph. So here these numbers, these rows and columns are indexed by these persons. So here this column is 1, 2, 3, 4, 5 indexed by these persons. Rows are also indexed by these persons. If they are friend with each other, then the entry will be 1, otherwise entry is 0. Okay, so 1 is not friend of itself, so this entry is 0. 2 is friend of 1, so this entry is 1. 1 is not friend with 3, so this entry is 0. 1 is friend with 4, so this entry is 1. 1 is not friend with 5, so this entry is 0. Similarly, 2 is friend with 1, 3, and 4. So 1, 3, and 4. 3 is friend with 2 and 5, 2 and 5. 4 is friend with 1, 2. Okay, all other entries are 0. 5 is friend with 3, so all other entries are 0. Okay, now I'm computing square of this matrix. So this is a 5 cross 5 matrix. I'm multiplying this matrix with itself. So in result, I'm again getting a 5 cross 5 matrix. Now this matrix has very important information about your friend circle. Can you guess that what that information this matrix A square has? So look at this matrix and graph for a couple of minutes and try to tell me what is the relation between them. Okay, so let me help you that from where to start thinking. So these entries along this line is called diagonal entries. Okay, so can you think of what is the meaning of diagonal entries? What is 2 signifies for 1, for this person 1, and this entry is 3 for 2, entry is 2 for 3. Can you see something? 4 is, for 4 it is 2, for 5 it is 1. Yeah, number of friends. So this diagonal entries tells, thank you very much. So this diagonal entry is telling me that 1 has 2 friends, 2 has 3 friends, 3 has 2 friends. You can see here 2. Now even these entries has a meaning. Okay, so let me tell you what this entry means. So this entry is 1, meaning 1 and 2 has 1 mutual friend. So 1 and 2 has 1 mutual friend, which is 4. Okay, now this entry is 1, that means 1 and 3 has 1 mutual friend, that is 2. This entry is 1, means 1 and 4 has 1 mutual friend, which is again 2. And this entry 0 means 1 and 5 has no mutual friends. So similarly, the other entries. So these off diagonal entries are telling you the mutual friends between those 2 persons. Okay, so this is how the Facebook uses the matrices. So if you compute a cube, that tells you your friends which are friends among themselves. So so basically Facebook use powers of these adjacency matrix to give you suggestions that you may know these peoples, and it also helps Facebook to compute your number of friends and your number of mutual friends, etc. Okay, I hope this is fine with you. So let's move to the second motivation for matrix multiplication, and please pay attention because this will be used later too. Okay, thank you, Priya. So here, here R means set of real number, Rn means n tuple of real numbers. So here I have a n tuple containing real entries. Now if A is n cross n matrix, then this X can be viewed as, so this should be capital X, I'm sorry. This X can be viewed as n cross 1 matrix, writing this way. Now I have n cross 1 matrix. This is, sorry, n cross n matrix. This is n cross 1 matrix. So because number of columns here is same as number of rows here, so I can multiply them. So resultant AX will again be n cross 1 matrix. So this belongs to Rn. So basically, when you multiply a element of Rn with A, the resultant will be again an element of Rn. Okay, so Rn, so any n cross n matrix behaved as a transformation on Rn. So a 2 cross 2 matrix will behave as a transformation on R2, and R2 you can look like a plane. So this is a rotation matrix. What do I mean by rotation matrix? So if I take vector here xy, and I multiply it with this matrix, the resultant will be rotation of xy by angle theta in counterclockwise direction. So if you are not convinced, let me do one quick example for you. So suppose we are taking theta is equal to 90 degree. If you remember trigonometry from your high school, then this is cos 90 is 0, this is minus 1, 1, sin 90 is 1, this is 0. So suppose I have here 0.10. Let's see what happened to 10. If I multiply, this is 0, here it is 0, then I'm getting 1. So it is going to, sorry, 0, okay, then 1. Okay, so 1, 0. One zero is getting mapped to 0, 1. So 0, 1 is here. So it is rotated by 90 degree in counterclockwise direction. So this is a rotation matrix with angle theta. This is rotation matrix with angle phi. Now, if I say you that I'm applying xy here, so this matrix will rotate this by angle phi in counterclockwise direction. Then if I multiply this with the next matrix again, so let's do what is happening. So suppose I have a vector over here. When I multiply with this first matrix phi, it will get rotated by angle phi in counterclockwise direction. When I again multiply it with rotation matrix with angle theta, it will further rotated counterclockwise in counterclockwise direction by angle theta. So the product of these two matrices should rotate this vector by theta plus phi. So which is true if you use trigonometry law and matrix multiplication. So matrix multiplication respects the multiplication of transformations in Rn. The matrix multiplication we have is same as multiplication of transformations in Rn. I have not gone in detail here, but from this rotation matrix, I think it is clear. But this will not happen if we multiply the matrices by coordinate wise. So the coordinate wise multiplication of matrices won't be useful for Facebook. Coordinate wise matrix multiplication won't respect the multiplication of transformations. That's why we don't multiply matrices coordinate wise. So the way we multiply matrices is same as the way we will be multiplying the corresponding transformations. Okay, and it has other benefits in technology as well. Okay, so now since we have learned matrix multiplication, we have learned how to add two matrices, we have learned scalar multiplication of matrices. This helps me to write system of linear equations in matrices form. So here this is a system of linear equations. So here this A is M cross N matrix, okay, having value Aij. So basically this is matrix made from these coefficients of these equations. That's why it is called coefficient matrix. And this is X is matrix of variables. It is x1, x2, xn. And B is matrix of these constants. So now if you apply matrix multiplication, scalar multiplication, then matrix addition, so basically AXB will be same as this system of linear equations. So using these three operations on matrices, we can write system of linear equations in term of matrices. That is how computer sees a system of linear equations. And this new matrix, this matrix A is called coefficient matrix, and we'll also form a matrix of this type. Here we have matrix A, and we have one more column which is coming from these constants. This is called augmented matrix. Okay. Now, once we convert system of linear equations in matrices, the various properties of matrices help us to solve this system of linear equations. So the first property we use here is rank of a matrix. To define rank of a matrix, I need to define row echelon form of a matrix. A matrix is said to be in row echelon form if all rows consisting of only zeros are at the bottom of the matrix. Here is no row, but if there is a row containing 0, that will be at the bottom. The leading coefficient of a non-zero row will be on strictly right-hand side of leading coefficient of row above. So leading coefficient, non-zero leading coefficient of this row is on the right-hand side of leading non-zero coefficient of this row. Okay. If we have a matrix in this form, then we'll say that matrix is in row echelon form. The number of non-zero rows in a matrix in its row echelon form is called the rank of matrix. So to compute rank of a matrix, we need to convert a matrix in its row echelon form. So how do we convert a matrix into row echelon form? For that, we use elementary row transformations and elementary column transformation. Actually, for elementary row echelon form, we only need row transformation. What do I mean by row transformation? We can interchange two rows. We can multiply any row with any non-zero number, and we can multiply with a row with a number and add it to another. So we use these three transformations to convert a matrix into row echelon form, which gives us rank of the matrix. So just to explain you, let's compute rank of this matrix to convert it into row echelon form. So the first thing I'm doing, I'm interchanging these two rows. Now I need to make this entry 0. For that, what I'm doing, I have changing R3 to R3 minus 5 times R1. So I'm getting 1 minus 1, 0, 0 minus 1, 0. 5 times this is 0. 3 minus minus 5, this is minus 8. Here I have 1, sorry. Okay. Then to make this entry 0, I change R3 to R3 plus, no, minus 8 times R2. So here I get 1 minus 1, 0, 0 minus 1, 1. And this gives me 0, 0. So now this is in row echelon form. In row echelon form, it has two non-zero rows. So rank of this matrix is 2. Okay. Now how ranks helps us in solving system of linear equations. So this is the method. So the first step is compute rank of coefficient matrix A and rank of augmented matrix AB. If these two ranks, here it is rank, not ran. If these two ranks are not same, then the system of linear equation has no solution. So we'll stop here. If these two ranks are same, then there are two possibilities. One is either system of linear equations may have no solution, or it may have infinitely many solutions. So if these ranks are equal to number of variables, then system of linear equation will have unique solution. And if this rank is not equal to number of variables, then it will be having infinitely many solutions. So we'll see this method with couple of examples. So this system we have already seen, it has a unique solution. So here, if you write matrix AB, that is 1 minus 1, 1, 2, 1, 1, 1, 4, 1, 1, minus 1, 2. If you'll compute rank of A and rank of AB, you will get all these, both ranks are equal to 3. Here number of variables are also 3. So by this matrix method, it has a unique solution. Okay. Rank of A is same as rank of augmented matrix, which is same as number of variables. That gives you unique solution. So this has unique solution in that case. Okay. So here, if you write this system of linear equations in matrix form, we have 5, 3, 8, 17. 0, 1, 1, 2, 1, minus 1, 0, 0. Try to compute its rank. Okay. So 5, 3, 8, 17. 5 times this, I'll minus, or I can change these two. So if you solve this, you will get that rank of A is 2, but rank of augmented matrix is 3. So because rank of A is not same as rank of augmented matrix, that's why there is no solution in this system of linear equations. Okay. And this is one example of infinitely many solutions. Here, there is a slight change from the previous example. So this is matrix A, and the total one is augmented matrix. Here, if you compute, then you will get rank of A is same as rank of AB, and both ranks are equal to 2. Since both ranks are equal to 2, which is less than number of variables, because number of variables is 3. So in this case, it has infinitely many solutions. Okay. And if you solve this rank, that will also help to get that what are those infinitely many solutions. So with this discussion on system of linear equation completes. So matrix method helps us to determine whether what type of solution of system of linear equations has, and it also further help us to get the solution if it exists. Now we go back to the image processing. So we know that a computer stores an image as matrix. Okay. And we want efficient methods to store a image in computer, that it should not take too much memory of our computer. So for that, we sometimes compress the images. So the way we want to compress the image is that we want to retain the quality of image, but we want it to take less space on the computer memory. And this is achieved by doing singular value decomposition of the metric associated with picture. Okay, so what is the singular value decomposition? It is the factorization of that matrix into 3 matrices. Okay, and how do we obtain those 3 matrices? I'll explain in a minute. This decomposition has many important applications in many algorithms of data science, machine learning, etc. And it is very important in image processing too. But to understand the mathematical idea behind singular value decomposition, we need to understand eigenvectors of a matrix. So let's see what is eigenvector of a matrix. So we have seen that matrix behaves like a transformations on Rn. Okay. So now suppose I have an element in Rn, which is x, and I have Ax is again in Rn. Now, how x and Ax are related with each other? That will basically depend on the matrix. Let's take example of these 3 matrices to explain that how it depends on a matrix, that how a vector and Ax is related. So if I have this rotation matrix, okay, so we know that how this x, and if this is A, x and Ax are related. So basically Ax is rotated by angle theta in anti-clockwise direction. Okay. And how this matrix will behave on xy? This is giving me kxy. So basically, if you have the vector, say 1, 0, 1, 0, I'll multiply here, I will get k0. So this vector 1, 0, which if k is greater than 1, then on multiplication with this matrix, it will get stretched to K0. So this matrix is stretching vector along x-axis, and the matrix below it, this matrix, this is stretching vector along y-axis. Similarly, if you are taking now 0, 1, and you are multiplying with this matrix, and suppose k is greater than 1, then it is getting stretched along here. If k is less than 1, it is getting shrinked along here. So these matrices here, this is rotating vectors. This is basically the scaling vector. But if you are taking arbitrary matrix, its effect on a vector will be mixture of rotation and scaling. If I multiply a vector with an arbitrary matrix, that vector will be rotated as well as it will be shrinked or stretched as well. Okay. So effect of a matrix on a vector depends on vector too. Okay. So it is possible for a fixed matrix, you can find a vector x where ax is just a scaling of that vector with no change in direction. For example, this is an arbitrary matrix. Okay. If you are taking any arbitrary vector, its result on that vector will be mixture of scaling and stretching. But if I'm taking vector 1, 1, the answer I'm getting is 3, 3. Here, the direction of this vector has not changed. It only got stretched. So I have this vector 1, 1. On multiplication with this matrix, I got answer 3, 3. So direction has not changed. It only got stretched. So such vectors which on multiplication with matrix, such vectors whose direction does not change on multiplication with matrix are called eigenvectors. And they play a very important role in image processing, as we will see in coming slides. Okay. So any question till now? Any questions? Okay, if no, then I'll continue. So what is definition of eigenvector? Let A be a n cross n matrix. A non-zero vector x is called eigenvector if ax is equal to lambda x, and lambda is called the corresponding eigenvalue. Okay. Now what is the geometrical meaning of eigenvector of 2, 1, 1, 2? So suppose this is unit circle. Okay. This is matrix A. Now I am taking any vector which lies on this unit circle. Then how AT will come out? So AT will be, this is 1, 1, which is one eigenvector of this matrix. And this is 1 minus 1, which is another eigenvector of this matrix. So it will get, okay, so to make complete sense, let me tell you. So this is 2 cross 2 matrix. It has two eigenvectors. One is 1, 1 corresponding to eigenvalue 3. This is, if you multiply, two times minus 1, you are getting 1. So this is again eigenvector corresponding to eigenvalue 1. So suppose this is 3. So this circle, if this is circle, then if this is vector t, this will be AT. So this circle will get converted to an ellipse. And it will get stretched in the direction of these eigenvectors. And the stretching corresponds to eigenvalue. Larger is the eigenvalue, more is the effect of this matrix. Because here eigenvalue is just 1, so it won't stretch in this direction. Here eigenvalue is 3, so it will get stretched in this direction. This is how the eigenvectors play an important role. Okay. So this matrix has very important property that we'll see after this definition. A matrix, for a matrix M cross N matrix, this is M cross N matrix. We define transpose of matrix by making rows into columns of this matrix. So this is the first row of this matrix. When I'm taking its transpose, this first row will become first column. The second row will become second column. Continuing so on, mth row will become mth column. So transpose of m cross n matrix will be n cross m matrix. And a matrix if it is same as its transpose. So this matrix which we are discussing, if you take its transpose, you will get same matrix. So this is a symmetric matrix. Okay. So please note that in the definition of symmetric matrix implies that all symmetric matrices are square matrix, because A is equal to A transpose. That means both are n cross n matrices. Okay, square matrix means number of rows is same as number of columns in that matrix. Symmetric matrix has very interesting properties. All the eigenvalues of symmetric matrices are real numbers. So it is not possible that all eigenvalues of all matrices are real numbers. I'll give you examples. Example of a matrix in coming slide which does not have real eigenvalues. Okay. And in case of symmetric matrix, we can find an orthonormal set of its eigenvectors. So what do we mean by orthonormal set? I hope you understand the dot product between vectors of Rn. So suppose u, v are two eigenvectors. If they are distinct, then their dot product will be 0. And if dot product of u with u is 1. So a set of vectors having this property is called orthonormal set. So for a symmetric matrix, we can find a set of vectors. We can find eigenvectors which forms a orthonormal set. So let A be a symmetric matrix. Then there exists a matrix P whose columns are eigenvectors of A. And because these are orthonormal set, so this has this property that dot product of eigenvector with itself is 1. And it is 0 if we are taking two distinct eigenvectors. So this implies that P is invertible matrix and inverse of P is same as its transpose. And if you multiply these three matrices, P transpose with A and again with P, the resultant is a diagonal matrix. What do we mean by diagonal matrix? A matrix whose only non-zero entries are diagonal entries. All other entries are 0. Okay. And these diagonal entries will be precisely the eigenvalues of A. So these are the properties of symmetric matrix. So you can, this we say in mathematical language that you can diagonalize a symmetric matrix by a orthogonal matrix. Okay. And this leads to eigen decomposition of symmetric matrix. So just now we have discussed that a symmetric matrix is orthogonally diagonalizable. So there exists, if A is n cross n symmetric matrix, then we can decompose it as A is equal to PDP transpose, where D is n cross n diagonal matrix and diagonal entries of this diagonal matrix are nothing but eigenvalues of A, and columns of P are n linearly independent eigenvectors of A. So we have this situation. So this is a matrix P. This Ui is eigenvector of A. This is matrix D, diagonal matrix. These diagonal entries are eigenvalue of A. And here we have P transpose. If you multiply these three matrices, you are getting this equation. Here U1 and U1 transpose is again n cross n matrix and it is multiplied by lambda 1. So these Ui and Ui transpose are called projection matrices. So we can decompose matrix A into, if A is n cross n symmetric matrix, we can decompose it into n projection matrices. Okay, and what is use of this decomposition? The eigen decomposition breaks n cross n symmetric matrix into n matrices multiplied by one of the eigenvalues. The eigenvalues play important role as weight of these matrices. Bigger is the eigenvalue, more is the weight of that projection matrix. So we can approximate our original symmetric matrix by summing projection matrices which corresponds to bigger eigenvalues. Okay, so we arrange these eigenvalues in descending order. So lambda 1 is greater than equal to lambda 2, which is greater than equal to lambda 3, and so on. So we can approximate matrix A by taking first k, sum of first k projection matrices. So I'm explaining this with the example. It's a very small example. I'm just taking 2 cross 2 matrix. So this is a 2 cross 2 matrix. If you compute eigenvalues of this matrix, you will be getting 5.2059, and lambda 2 you are getting 0.1441. So there is huge difference between these two eigenvalues. This is 5.2 and this is just 0.1. So corresponding eigenvectors, if I compute, I'm getting u1 and u2. Eigen decomposition is lambda 1 u1, u1 transpose, and so on. Now, if I approximate this matrix with just first projection matrix, I'm getting this matrix. Now you compare this matrix A1, which is coming from just taking first part. So we are ignoring this second part, lambda 2 u2 and u2 transpose. We are just taking lambda 1 u1, u1 transpose. So here you see, if you compare these two matrices, the given original matrix and this matrix. Okay, so first entry here is 5, here it is 4.9934, here entry is 1, here 1.02. So it is quite close to our given matrix. So because there was huge difference between these eigenvalues, so ignoring the projection matrix corresponding to smaller eigenvalue has not given much effect on our matrix. So what is use of doing this approximation? So here this A 2 cross 2 matrix will contain it has 4 pixels. So we need 4 units to store this. Here we need only 3 units to store this. U1 has 2 values and lambda 1 is 1 value. So I need only 3 units to store this vector. This approximation, because here I'm taking this just 2 cross 2 matrix, so this difference is quite less, just of 1 pixel. Suppose if you have a matrix which is having million of rows and then you are approximating it with say 30 or 50 eigenvalues, then there will be a huge difference in the storage space that we are there will be a huge saving of storage space by saving approximated matrix instead of the original matrix. I hope this is fine with you. Okay, so this is usefulness of eigen decomposition. We can approximate n cross n matrix. Suppose this is n cross n matrix, so this will take n square pixels to store, but this approximation will take, so suppose we are taking till k, so this will take, sorry Priya, what are you saying? I missed your comment. Sorry, can somebody read comment of Priya? Okay, maybe you can ask later. Let me finish what I want to say. So here I need n square spaces to store this matrix. If I approximate it with first k values, then I need less space to store this matrix. So now next question is that we know that we can find eigen decomposition only for symmetric matrix. So we can't find eigen decomposition for all matrices. This is the example of matrix which has no real eigenvalues and it has no eigenvectors. So if you want, you can say this is A and you can try to solve this equation and you will end up of finding no non-zero xy which satisfies this equation. So that will prove that this matrix has no real eigenvalue. So we can do only eigen decomposition only for symmetric matrices, but we were saying that we want to compress images. So usually images are not even square matrices. Forget about the symmetric matrices. So how do we use this idea for images? For that, we need to modify this idea. There the singular values comes into play. Okay, so suppose A is M cross N matrix, which is definitely not symmetric because this is not square matrix. Then you multiply this matrix with its transpose. The resultant matrix is a symmetric matrix. So transpose has this property that AB transpose is same as B transpose and A transpose. Using this, you can check that this matrix is a symmetric matrix. Every symmetric matrix has a real eigenvalues. So, but for this matrix, which is A transpose A, even more is true. It has not only real eigenvalues, all the eigenvalues are positive numbers as well. Because all eigenvalues are positive numbers, so we can take it's a square root. Okay, so the positive square root of an eigenvalue of A transpose A are called singular value of matrix A. So what we are doing, we have matrix A, we are computing A transpose A. This has positive eigenvalues. We are taking square root of these eigenvalues. This is called, this will be written by sigma. This is called singular value of A. Okay. And if rank of matrix is R, then it will have R singular values. Counting with multiplicity means that sum of singular values may be same. Okay. Now, if A is M cross N matrix, rank of this matrix is R. It's a singular value decomposition is decomposition of this matrix into 3 matrices. First, we are writing with U, second is with sigma, third is V transpose. Now, what is V? The columns of V are normalized eigenvectors of symmetric matrix A transpose of A, which are orthogonal to each other. And sigma is a diagonal matrix, M cross N diagonal matrix, whose first R diagonal entries are singular values of A and all other entries are 0. And U is obtained from the first R entries of U are AVI divided by sigma I. Okay, where VI is eigenvector of A transpose A corresponding to eigenvalue lambda I, and square root of lambda I is same as sigma I. And other vectors are obtained using Gaussian elimination, Gaussian orthogonalization process, which I'm not going to discuss. So in short, I want to say that in singular value decomposition, you write A matrix as product of these 3 matrices. Okay, and so in first you have orthonormal vectors U1, U2, Um. This in between if this is M cross N matrix, this diagonal matrix is also M cross N. Its first R diagonal entries are singular values of A and all other entries are 0. Here these are coming from normalized eigenvectors of matrix A transpose A. So here we are writing A as sum of these projection matrices. Okay, and again, like eigen decomposition, to save the space, we can approximate it with lesser number of projection matrix. So here is the example. I quickly go through it. If A is this 3 cross 2 matrix, we compute A transpose A, which is 2 cross 2 matrix. We compute its eigenvalue, take square roots to compute singular values of A. Then we find orthonormalized eigenvectors of A transpose A. This gives us matrix V. Okay, so here is this matrix V. This is matrix sigma. Okay, and here we are computing U, and we got this decomposition. You can multiply and check this is same as A. Okay, now what is use of this approximation? For that, I want to show you the small program which I made on Sage. I want to stop sharing. How to do that? Okay, so this is example of SVD in image processing. I want to show, sorry. Okay, good. I got how to stop sharing. Okay, so I want to share another file now. Yeah, here it is. So this I have obtained using a computer software known as Sage Math. Sage Math is open software. That means anybody can install this software. So here it is using SVD for image processing. So it's using PyLab and NumMe. So I am basically read that matrix, that image. So here on my desktop, I have saved this image of a building in IIT Jodhpur. When I asked to plot this image, plot this matrix, because it's storing image as matrix, I got this is image. Okay, and here this shape of image tells me that I am having 183 pixels along the height of this matrix and 275 pixel along the width of this matrix. 3 says that this is a colored image. Okay, and then first I'm converting it into grayscale image. This is a standard formula to convert an image into grayscale image by multiplying R with 0.299, green with 0.587, and blue with 0.114. So we convert this image into grayscale image. Now next we are computing. So here it is shape. Now 3 is gone because it is a grayscale image. Next we are computing USV, which is SVD of this matrix, the matrix corresponding to this grayscale image. So you can see the dimension of these matrices. Okay. So now we are in first attempt, we are approximating it by considering just first 5 singular values. So it will have at least 183 positive singular. So it will have at most 183 singular eigenvalues, singular values. So we are in first approximations, we are just taking first 5 eigenvalue, first 5 singular values, projection items. So I'm getting this very blurred image. So as I increase number of projection matrices I'm considering, here I'm taking 10 projection matrices. So this is somewhat clear, better than the previous one. Then I'm taking with 30, it's still better. And finally I have took with 50, which is quite close to the original gray image. Okay. So this uses SVD for image processing. Here is the result. So this original image is having 183 cross 275. So total number of pixel is 50,325. So the next slide shows you its approximation with 50 singular values. So the total number of pixels, sorry, here total number of pixels becomes 22,000, which is less than the 50% of the original one. Okay, so if you want to learn, so basically this is the last thing I want to discuss. So this uses a singular value decomposition for image processing. Any questions? So if you get interested, you can see these 2 articles online. They talk about mathematics in a very user-friendly language. So this one article, A Compressive Beginner Guide Linear Algebra for Data Science. So any even our non-mathematical students can easily read these 2 articles to gain further knowledge on this topic. I stop here. Thank you very much. Any questions, please? Here is a disclaimer that I'm not doing research in image processing area. What will be the dimension of SVD? So SVD is compressed in SVD. we are having 3 matrices. If you are doing SVD of M cross N matrix A, suppose to start with A is M cross N matrix, then in SVD you are getting 3 matrices U, sigma and V. So dimension of matrix U is M cross M, dimension of matrix sigma is M cross N, dimension of matrix V is N cross N. Okay. So I was saying that I'm not expert in this area of image processing. So if I have made any mistake and you notice it, please feel free to share that with me. I thank you very much for Pankaj. I thank you very much to Pankaj ji for giving me this opportunity, because of this I learned a lot. I use Sage Math for my research work, but before preparation of this talk, I didn't know that I can also do image processing using Sage. So that was quite new thing for me to learn in the preparation of this talk. So it was quite a good learning opportunity for me as well. Thank you very much, Pankaj.
Thank you, ma'am, for enlightening us with your remarkable and engaging talk.
Okay, I can stop here now, Monica.
Yes, ma'am.
Okay, thank you.
Any more questions?
Thank you, Priya.
It gives me great pleasure to address this closing session for today. Allow me to first convey special thanks and appreciation to Dr. Dilpreet Kaur, ma'am. I thank all the participants who have attended the session. See you all tomorrow. Thank you.
Thank you, everyone. Thank you very much. Thanks, Monica. Thanks, Pankaj. Bye.